Good afternoon, everyone, and welcome. My name is Jamie Williamson. I'm Executive Vice President here at Scripps, and we're here to honor an extraordinary man, Richard Lerner, and all of his contributions to science and to Scripps. So we'll have an afternoon of scientific talks, but we want to kick it off with a tribute from none other than our President and CEO, Pete Schultz.
Thanks, Jamie. If I could have, I'm going to not talk about science, my science. I'm going to talk about Richard, who was pretty amazing. The biggest problem, I was just talking to the speakers, the biggest problem we had was deciding which of the Richard stories we could tell and which ones we couldn't, okay? So I'm going to recollect on Richard's career and some stories to give you an insight into Richard's persona. Richard grew up in Chicago, and he always used to tell me about his lifeguard stories on the beaches of Lake Michigan. And I think that kind of explained his affection for the beach and the ocean in San Diego. He got his MD degree from Stanford. And there's an interesting story there. He actually told me about the first kidney dialysis that he performed at Stanford. And a guy came in and he really needed a dialysis and they couldn't get a machine. And so the rule was, you don't dialyze the person. And Richard never cared about rules, okay? He just thought he should do what he should do. So Richard gets a bucket out and mixes all the buffers and starts dialyzing this guy. And all of a sudden the guy goes, Dr. Lerner, and starts shaking and passes out. And Richard checks the pH of the buffer, buffers it. Guy wakes back up and does this 3 times, okay? But Richard kept pHing the dialysis buffer and actually wound up saving the guy's life and he got to dialysis, but he incurred the wrath of the Stanford School of Medicine. But I think after that, Richard was like, I don't want to be a doctor, okay? So I'm going to be a scientist. And he started his career as a fellow here at Scripps. And one of the first experiments he did with Frank Dixon was to really understand that Goodpasture's disease was an autoimmune disease and actually prove the underlying autoimmune basis for Goodpasture's disease. So he started his career on really a spectacular note. After he was done being a fellow here, he moved to the Wistar, but he was only there for about a year before he came back to Scripps and started his early lab here. And you can see Richard in the middle with all of his coworkers. And one of the first things Richard did was the development of synthetic vaccines. So he realized small peptide sequences could also serve as important antigens for the development of vaccines. And that was kind of the start of his independent career. And I would say, knowing Richard, you know, he was a remarkably creative person. He would come up with so many, every time I would talk to him, he would have a new idea. And every now and then, some of them were a bit over the top, but the beauty about Richard is he was always fearless. It was a new idea, and he thought it was good. He would just do it, and he didn't care what he had to learn and what he had to know to do it. And that was the nature of Richard's science and his whole career. And I first got to know him in the catalytic anti days. Richard was, I would say, at the very start, if not the start, of what's called chemical biology, a term he really didn't like. But he was one of the first to realize that you could seamlessly integrate chemistry and biology, in his case, immunology. And there was tremendous power and opportunity at that interface. And that was pretty remarkable for an MD to understand that. And he was also one of the first, if not the first, to realize the power of molecular diversity in chemistry. In his case, harnessing the molecular diversity of the immune system, something he knew well, the humoral immune system, combining that with fundamental notions of chemistry to create antibody catalyst. And again, that concept, I think, was the first real push of chemists into this idea of molecular diversity. And that was a theme that he followed his entire career. I met Richard because we used to have something called a Pacific Coast Lecture, and I think it was Albert Eschenmoser. And I was a second-year assistant professor at Berkeley, and I'd just done something I thought was clever and make a catalytic antibody for the first time. And Albert looked at me and he said, you know, I was just down at Scripps and I heard the same story from Richard Lerner. And as a second-year assistant professor with what you thought would have been the most important thing you were going to do, I was a little disconcerted. So I decided to call Richard. And, you know, Dr. Lerner, you don't know me, but I'm an assistant professor, second-year assistant professor at Berkeley. And, you know, Richard could have just overwhelmed me, okay, because he had all of the resources of Scripps. But he didn't do that. He actually, he said, how can I help? And I'm like, what do you mean? And he said, well, I can start making antibodies for you if you like. I'd like to work with you. And so that kind of captured Richard's spirit for science and his generosity. And so that was my first exchange with Richard. And then Richard went on, learned organic chemistry again. You would always see him with Morrison and Boyd or some mechanistic organic chemistry. And he put together a group that included Kim Janda and Carlos Barbas and Steve Bankovic and others. And for the next 15 years, just published a tour de force at kind of the interface of chemistry and immunology. And it kind of, a crowning achievement was he developed an antibody that catalyzed an aldol reaction very selectively. And that formed the basis for COVAX, which developed that into human therapeutics and was very successful. It was acquired by Pfizer for quite some sum. And Richard, you know, that Richard did have an entrepreneurial spirit, and he was involved in many boards and many biotech companies and very, very successful. And that success actually enabled another hobby of his, which I'll call his collector gene. Richard used to always collect everything, okay? And then he got into watches, and he really started collecting watches. So he came in one day, and Pete, let me show you this. So he has on a Patek watch. And he's like, look at this. And I looked at it, and he said, you need to go get one. And I'm like, I think it's a $200,000 watch, Richard. I'm like, no. So a few years later, he comes in and he says, Pete, I have a gift for you. And I said, what's that? And he said, he pulls out a Patek watch. And I'm like, you gotta be kidding, Richard. There's no way I'm gonna take this from you. And he goes, don't worry about it, don't worry about it. I picked it up in Shanghai. It's a fake. I paid $50 for it, okay? You can't tell it's not a Patek unless you open it up, okay? So this gets to a Marty Friedlander story. So Richard and Marty are at a conference walking along a lake. And, you know, Richard's reputation in biotech and the venture community was well known, so young venture capitalists ran up to him and said, Dr. Lerner, I really wanted to meet you. You know, I'm just hope my career can be as successful as yours, and one day I'll be able to wear a Patek watch like you. And Richard takes off the Patek watch, throws it in the lake, and says, "Good luck." Guy didn't know it was a Shanghai watch. Okay. So then Richard, took this idea of diversity and was one of the very first people to make combinatorial libraries of antibodies, okay, in phage, and really was the pioneer in that area. That work formed the basis, the intellectual property basis for Humira, which has impacted the lives of very many people around the world. Richard also, as a chemist, was the first one to propose the notion of DNA encoded combinatorial libraries, along with Sidney Brenner, which is a close friend and collaborator of Richard's. And today, encoded combinatorial libraries, DNA encoded combinatorial libraries, are having a huge impact, and many people use them all over the world to do basic research and make new drugs. And then unfortunately, Richard suffered a stroke. And when you go visit him, he still talks science. He talked a little slower. In fact, I would argue that was the only time in his life where there was a little slowness between his brain and what he said, okay? But he functioned at the highest levels even with a stroke. I mean, he published a really important paper on the mechanism of general anesthesia. And he would sit there and talk to me about it, and it was just terrific, okay? How many people have the capacity, even after a major stroke, to do science at that level? He was amazing. But there are some publications we don't know about, okay? Epidemic 9, okay? So Richard, I'm talking to Richard about nonfiction, about fiction horror stories, and he goes, "Well, I wrote a book." And I'm like, "No." And he goes, "Yeah, Epidemic 9. And I'm like, and what's special about that?" And he said, "It has the best chapter on an autopsy you've ever read in fiction." Okay. Richard's contribution, okay. And as you were watching the screen up here, you realize Richard, did just spectacular and one of the most creative, daring scientists I've ever known. And he won a lot of awards. Here he was with the Paul Ehrlich Prize, Germany's highest award, the Asturias Award, the Wolf Prize. The Wolf Prize, I got together with Richard and kind of just use it as an example of Richard. What was fun about Richard is he wasn't always politically correct, okay, usually in a humorous way. And he really had a complete disregard for convention. So when we got the Wolf Prize, Udi can attest to this, I had to give the first speech the night before because I was a young person. So I gave what I thought was a pretty good address. Richard, what do you think? Because I was talking for both of us. Yeah, that was pretty good, Pete. And so, what are you going to say? Oh, I got it all figured out. Richard, what are you going to say? Okay, because this was in the Knesset with, on live TV across the state of Israel. So I'm kind of like, Richard, can you tell me? No, I got it. So he gets up there, and I'm looking at Nikki in the audience, and I'm like, Nikki, I'm not sure what he's going to say, okay? So Richard gets up there and says, I'd like to thank you on behalf of myself and Pete for the Wolf Prize. And then he said, have you heard the one about the chemist, the biologist, and the rabbi? And I'm looking at Nikki, and Nikki's kind of going like this, okay? And he goes, they were arguing about what was the most important scientific contribution of the 20th century, because this is a Wolf Prize. And the biologist says the structure of DNA. And the chemist goes, no, the quantum mechanics. And the rabbi goes, no, you guys are both wrong. It's the thermos bottle. And I'm like, what? And everybody who's in the audience is going, what? And he goes, yeah. You put hot water in it, and it stays hot. You put cold water in it, and it stays cold. How does it know? That was Richard's acceptance speech for the Wolf Prize. I loved it. And then we got an award together once from Siva Geigy, okay, and it was given at a small kind of biomedical undergraduate research school in the East Coast. So I fly in that morning, I take the red eye, Richard is there for the dinner the night before, and I walk in at 7:30 in the morning, and Richard's got his suitcase walking out. And I'm like, wait, Richard, I thought the ceremony was in an hour. And he goes, yeah, I'm leaving, Pete. And I go, what do you mean you're leaving? We got to be here, it's in an hour. And he goes, well, to get the award, we have to give a talk to the entire senior class. And I told them, I'm president of Scripps. The reason we don't have undergraduates is because I don't like doing this, okay? I'm like, Richard, don't worry about it. I'll teach sophomore organic chemistry, I'll do it. Okay, Pete, if you do it, I'll get up there, okay? So we get up into the auditorium, this huge amphitheater with, the whole entire senior class. And they got a lectern like this, and the whole class, and our host calls up Richard and says, Dr. Lerner, why don't you start? And Richard looks at me, and he's like, Pete. And so I'm like, Richard. So he gets up to the lectern, and he sits there and doesn't say a word for 5 minutes. And the host is getting really nervous, and the students don't know what's going on. And finally, Richard goes, well, if nobody has any questions, why don't I start? Why don't I tell you why I'm president of Scripps? It's because I want to ensure that we don't have undergraduates and only graduate students, okay? The host was just, and the students didn't know what to say, but I knew Richard, so I started laughing. And then everybody else started smiling, okay? And then somebody got bold and said, well, why did you decide to be go into medicine? And he said, I didn't have the manual dexterity to be a car mechanic, okay? So this thing goes on for an hour, and they couldn't stop people from asking Richard questions, okay? I didn't even get up there, okay? But it was typical Richard. He was the best speaker in the whole series that they ever had. The students loved him, okay? And then I would travel every now and then with Richard, and it was always an interesting experience, okay? I'm not going to tell you about the Concorde story, but I was with him coming back from Boston once, and he and I weren't seating together, and he sits down next to somebody next to him, and he starts looking at what the guy's doing, and he says, oh, are you a chemist? And the guy goes, yeah. And he goes, where are you from? And the guy goes, I'm in Harvard chemistry department. And Richard looks at him, and the guy looks at Richard and says, where are you from? And he goes, I'm from Scripps Research. And what do you do? Oh, I'm an administrator, okay. And the guy looks at him, and he goes, well, do you know Richard Lerner? And Richard goes, why? And he said, well, I'm the first person to get tenure in 25 years at Harvard, and I just made a highly successful company, and I'm wealthy, and I want to ask Dr. Lerner what I should do next. So this person didn't realize he pushed 2 wrong buttons with Richard. He was self-congratulatory, and he was from Harvard, okay? So Richard looks at him, and he goes, I've met him once, but he's a really scary guy. And he really doesn't like Harvard professors, okay? So this guy got off the plane after 5 hours and still didn't know he was sitting next to Richard Lerner, okay? So Richard's like, well, let me tell you, Pete. So I went back to my office and I called this person and I said, I heard you sat next to Richard Lerner on the plane. He just about had a heart attack. But that was Richard. He could be really fun and kind of irreverent and a kid sometimes. He loved pursuing new opportunities and building bridges, okay? And Raymond will tell us a little about what he did with Oxford. But Richard was always looking for new and exciting things to do and bridges to build with the rest of the world. That was Richard. And while he was doing all of that, which would be more than enough for almost all of us to do with our lives, okay, and our careers, he was building Scripps into a scientific powerhouse. Okay, here he is at the 50th. So that's really remarkable. And Richard always pushed the frontiers, was never afraid of the unconventional. So how do you build a research institute? Where do you get the money? Let's get it from pharma. Let's do pharma deals, okay? At the time, it was not only unconventional, it was kind of controversial. Richard didn't care, okay? Nowadays, it's viewed as a real plus, okay, when you have a big pharma partnership. And not only that, you never said never to Richard, okay? Even though you might say, Richard, I don't think there's any way in hell that's going to work, okay? Richard always pulled the rabbit out of his hat. Who else could take a biomedical research institute and get Pittsburgh plate glass to give unrestricted money to it, okay? I mean, incredible, okay? And then he used that money to build Scripps, as I said, into a powerhouse. He had exceptional taste for people, both young, here's a young Chi Hui Wang, and established, Barry, okay? And he had the charm and the vision to convince these people to come to a relatively unknown institute, it's certainly from a chemistry perspective in La Jolla. And Barry told me the story about Richard recruiting him. And Richard would take people on walks all the way down to the shore and on Black's Beach and then back. And Barry's walking along with Richard, and up comes somebody on a beach cruiser bike with an igloo, carton, attached to it. And the guy is absolutely buck naked, okay, because it's a nude beach. And he pedals up to Richard. And Richard looks at him and says, Joe, how's my credit? And this naked person looks at Richard and says, Dr. Lerner, it's terrific. Well, fine. Could you please give me an ice cream bar for Barry here, okay? Now, how many presidents of major research institutes would do that, okay? I can't imagine the president of Harvard doing that. But the bottom line is Barry's here, and Casey's here, and Chi Hui's here. It worked, okay? And then here's a picture of a young Jeff Kelly and Paul, okay, together with Richard and Nikki. And Nikki was the angel who always whispered in Richard's ear when he was getting ready to cross the line, Richard, Richard. So thank you, Nikki. And then there was the one that got away. So Richard was at a board meeting, and he got into shotguns because they were shooting shotguns in West Virginia, I think. And at the time, I was shooting shotguns trap with my son. So Richard's like, oh, we got to start. We got to get into rifles. And I'm like, I don't know about that, Richard. So no, I got an idea. So he orders these really expensive German pellet guns that are basically like 22s, okay? And they come in, and he calls me, and he said, come up to my office. So I come up, and they're here, Pete. So he unwraps them, loads them with a pellet, and he says, let's try it. Where? So we go into his outer office in the EO, and he puts a phone book right next to the door coming into the outer office. And he said, let's see how many pages this will go through. So Pete, fire it. And I'm like, are you sure we should do this? And so I get over there at the other end, and I aim, and I fire. And right as I fire, the candidate, the assistant professor candidate we had that day, walks through the door. And here I'm firing a gun, okay? And I'm like, oh, what? Oh shit! What's Richard going to say? You know, this is a candidate for an assistant professor position. And I turn to Richard and I look at Richard and he goes, "You missed, Pete." So that person is now a very well-known professor in New York City, but he was out here recently and I was recounting that story and he was just laughing hysterically because he remembered it. Okay. Richard, why don't we build a graduate school? Why not? Richard, do you really think we can do that? Of course we can do it. That was Richard. No barriers, okay? So he built a graduate school with Ian and others that were here, okay? And it's been a top 10 graduate school for 20 years, and we just went through our 30th graduation yesterday, okay? Again, Richard Lerner, pioneering. And then the Skaggs partnership. Richard also enabled Scripps and brought people here through his relationship with Sam Skaggs, okay, bringing in well over $100 million into the Institute. And what was special about Richard is he didn't fund projects. He funded people. He just gave people the resources they needed to do their best science. And Barry even said, it gave me a second career here. And now we're actually on our way to endowing the entire Skaggs Graduate School. And Richard knew no barriers to science. He didn't care about politics. He didn't care about political correctness. Okay, if there was good science going on in China, he did science in China, okay? And he had a huge lab and influence on Shanghai Tech, which is a major research institute in China. And so here's a picture of Richard. I think Richard wouldn't have liked this picture, right, Nikki? Anyway, but it's the spirit that counts. And then really kind of Richard defined Scripps by the desert meetings, okay? And not only was the science there outstanding, but it was a really classy affair, okay, with an elegance associated with it, which really also reflected on Scripps as an institution. And everyone used to look forward to this. And one year I came up, this is my last story, one year I came up in my Volvo, and I pulled up to the hotel, and there's a beautiful turbo Porsche sitting outside the hotel, one of the old ones, Jeff, that was. And I walked in, and Richard goes, "Did you see that turbo Porsche?" And yeah, beautiful car. I said, "Do you know whose it is?" And he says, "Mine, I just bought it." And I'm like, wow. And he goes, "What'd you drive here, Pete? Your Volvo?" And I'm like, okay, that's a challenge, Richard. So I'm like, "Where'd you buy it?" There's this place called Symbolics in La Jolla. I said, "Do they have another one?" Richard and I used to always compete, but we competed in a way that was terrific. He was one of my closest friends, and he and I reinforced each other in an incredibly positive way. But this took the competition to a new realm. So I'm like, "Richard, let's get in your car and drive to Symbolics." "What do you mean, Pete?" "Let's just go." So we got in his car, left immediately, drove the 2 hours to Symbolics, and they had another turbo Porsche right there on the floor. And I said, "How much?" And it was a ridiculous amount of money. But I said, "Do you take credit cards?" And the guy said, "Yeah." And so I bought this turbo Porsche on a credit card so I would have my own turbo Porsche. So we drove those turbo Porsches back to Palm Springs at 150 miles an hour, okay? And that was Richard, okay? You only live once, Pete, okay? Go for it. So then one day I come in and all of a sudden this Porsche isn't out in the lot, it's a Ferrari. And I'm like, oh man. So Richard goes, "How do you like my Ferrari?" And I'm like, okay, let's go to Symbolics, okay? And I bought a Ferrari, okay? And then we're going to hit golf balls one Saturday morning, and it's raining, and Nikki didn't know this, but we did a 360-degree on the freeway, and Richard's Ferrari, okay? 5 days later, there was a blue VW Bug in Richard's parking spot at Scripps. And my Volvo was back, okay? But that was Richard, okay? You only live once, you know, go for it. So this next picture is my favorite picture of Richard, okay? Smoking a cigar on his porch in Jupiter, okay? And he really was just an amazing creative scientist. He was a builder, okay, that built one of the most remarkable research institutes in the world. Close friend and incredibly generous person, and he never feared doing something new and exciting, and really defied all of the boundaries we know, and really lived life to its fullest. So that's Richard.
All right, thanks, Pete. Thanks for what an amazing and fitting tribute to our colleague. Our next speaker, so the first scientific speaker, is Professor Phil Barron. Phil is a brilliant chemist. He is an amazing strategist and tactician of molecular gymnastics. He's a serial entrepreneur. He has many awards, including National Academy of Sciences and the MacArthur Fellow. Phil's chemistry is really, it's just very creative, and he develops new reactions and new methodologies that are both environmentally sensitive and efficient, that are widely adopted by the pharmaceutical industry and have really revolutionized the synthesis of medicines in the industrial sector. So he's a brilliant speaker. I don't know if any of you have seen his graduation address. He was the speaker at NYU at Madison Square Garden once. It's a must-see on YouTube. But today, Phil is going to give us a scientific talk. So please welcome Phil Barron.
Well, thanks so much, Jamie, for that super kind introduction. I feel really blessed to be speaking here today. And as a prelude to this talk, I want to tell you I believe Richard is here right now. Not maybe physically, but I really think he's in the audience at the moment. And so this talk is really for him. It is unconventional. As Pete mentioned, he was an unconventional person, and that goes with the attire I'm wearing today. You may be wondering, why am I dressed formally? I'm actually dressed more formally than any of you. So this jacket was given to me when Nicky was president, when he gave it to me a couple of years ago. Was given to him many years ago as an authentic Cerruti Italian jacket used by the Formula One team, Ferrari team. It's probably worth more than your car. And this watch, I believe this one's not from Shanghai. This one is also from Richard. The shirt I'll explain a little bit later. So this talk is interwoven with personal stories. It's supposed to be scientific, so it will be scientific. But before I begin, I think it's appropriate to tell you how I met Richard. So my first interaction with Richard in close proximity was on this stage 20 years and 4 days ago. That was a picture taken from this stage. Now, Artem gave a great commencement lecture yesterday, and he mentioned in it that he didn't remember his commencement speaker, and it might be because they gave a rather forgettable address. That wasn't the case when I graduated. Paul gave one of the most amazing talks I've ever heard, with no notes, no slides, just, I don't know how he did it, but he gave 10 minutes of incredible wisdom. And two of the quotes are shown here, one of which is, again, a testament to Richard. What he built through the graduate program at Scripps was a singularity that I think, I agree with Paul, will go down in American history as one of the turning points of graduate education. His belief that you should recruit the very best students and remove all of the bureaucracy that is associated with the conventional university and just allow them to do what they love to do, unencumbered, is still something that goes on today, that is our main competitive advantage over all schools out there. Another quote from Paul in his famous Back to the Future speech that some of you may remember is, I think, a thread that is woven into all of research that was done by Richard as well, and many of the faculty here, which is essentially always be trying to reinvent yourself and not resting on your laurels. I think laurels are best used as kindling rather than something, they're just uncomfortable. Why would you want to rest on them? And that was what Richard really taught everybody. I did include this video. This is pre-iPhone days, but I thought it would be cool to hear Richard's voice again.
As candidates for the degree of Doctor of Philosophy, you have submitted yourself to the traditional disciplines that have become the scholar in the pursuit of knowledge and have proved yourself worthy of this high calling. By virtue of the authority vested in me by the Board of Trustees of the Scripps Research Institute, I confer upon you the degree of Doctor of Philosophy. You may now move the tassels to the left side of your notebook.
Roughly 1 hour later, Richard gave me an offer to come back to Scripps. I'll never forget that moment, and the conversation went pretty much like this. Oh, hi, Professor Lerner. He said, oh, you know, I heard you're doing okay in your postdoc over in Harvard, which he had quite a bit of disdain, but yet respect for. And he said, I heard you're doing okay over there. And I said, yeah, everything is okay. And he said, well, if you can survive there, you can probably do well anywhere. So what's your plan? And I said, well, I'm thinking of applying to this school or that school. I don't know. And he said, well, why are you wasting your time with all that? Why don't you just come back? Well, you know, can you come back next week? I still had to finish my postdoc, but that was my offer. And to this day, Scripps is the only place that ever gave me anything, and any offer at all, on paper at least. So, and that was the extent of it. That was my first meeting with Richard. So today I'd like to intertwine the scientific portion with lessons that he taught me about life and science, and here are some things he taught me, and I suspect that he taught many of us that are in the audience today and online joining us. The first of which is on mentorship. And I think this is true even for the most sort of independent people we know. They can probably trace their inflection point in their life on a single person that made a really big difference and believed in them. Not just believed in them, but truly believed in them. It might be a parent, or it might be a friend, or in this case, and I think for many people here, it was Richard. He believed in people, and he celebrated them. So he was probably the one person that I could go to and share science, and he would rejoice as if it was his own result. So happy, truly happy, that you had accomplished something great as a sort of way to keep you motivated. But I think he truly believed that he was also, it was gratifying for him to see that he made a good decision to hire you. Back when I was being recruited as a graduate student in '96, the CNE News article shown here was given to all the graduate students. I had not met Richard then, but it explained what is some of the secret sauce of the Institute that Pete mentioned, which really is a lot about Richard. He was probably undeniable being the first person to recognize the problematic issue of siloing within the fields and the bifurcated nature between chemistry and biology. He noticed that you would have these two cultures that did not speak to each other, and reverberating throughout his science is a reoccurring theme of bridging the gap between fields. And he thought chemistry was a central science, and that if you could bring it together with areas of biology, magic would happen. I personally think chemistry was the subject he was probably most fond of, but others can confirm that. As Pete mentioned, he was also a master of seducing people to join the Institute. So I think many of you who were recruited by him, he had a way of recruiting that made you feel really special. And he also did it a lot by instinct. As Barry's quote here mentions, it's almost as if he sniffed it out. He could tell, like a hound dog, this person has got potential. And that was it. There was no bureaucracy, there was no main committee, hours of deliberations like choosing a pope. It was basically Richard thought this person's good, and that was it. Here's a recent picture from a few years ago. I think everyone that's seated at that table has amazing remembrances of Pete, and almost all of them were actually recruited by Richard to join the Institute. And that was a very special occasion. Okay, so the next thing he taught me was the issue of picking problems. And I think we heard about this from Pete as well, that not only did he like to think big, but he was very fond of getting people to steps way outside of their comfort zone. Getting them to, often you see folks, largely in more canonical research institutes and university settings, where they start studying something and they stick on that and they keep doing it for many years and they don't change. Richard cultivated an atmosphere where everyone was trying to reinvent themselves. When you roam the halls of Scripps and talk to the faculty, what is remarkable to me, and unlike any other place I visit, is that whether they're very, very advanced and won all sorts of awards, or they're just junior and starting, they all are acting like assistant professors without tenure. Tenure at Scripps is irrelevant. And that is the magic of the place he made. People always stepped outside their comfort zone. So let me give you an example of how stepping outside of your comfort zone for Richard could revolutionize the field of medicinal chemistry. So Pete touched on this a little bit, but I'm going to give you the detailed story of the origin and the development and exactly how it works in simple terms. So even if you're not a scientist today, hopefully you will understand what is the Dell dream that Lerner had back in 1992 to revolutionize medicinal chemistry. What is Dell? DNA-encoded libraries. And when he first came up with it with Sidney Brenner, I think it was poo-pooed by a lot of folks as being, a little bit off the wall, ridiculous, and probably of no pragmatic value at all. Fast forward 30 years, it makes the cover of CNE News as being one of the hottest trends in modern medicinal chemistry, and today has led to drug leads and probably even FDA-approved medicines that were discovered using this technique. But back in 1992, it was a bit bizarre, maybe even sacrilege, certainly very wacky. There's the paper. And as Richard tells the story, he was sitting with Sidney Brenner. They shared an office, and they were ruminating on the differences between chemistry and biology, when all of a sudden an organic chemist that he doesn't name walked in and started complaining about the problems that they had in reproducing a particular reaction and getting a high yield. This person was myopically focused on getting low yields. So I'll return to that in a moment. What is Dell? The magic of Dell is basically merging diversity of chemistry with the information of biology. What do I mean by that? Well, in organic chemistry, you've got reactions. That is, you go from compound A to compound B, and something needs to happen. And yield in an organic chemist vernacular is probably the foremost, most important thing you need to consider. If you're making a drug or a useful material or a natural product, whatever it is you're making, if the conversion only gives you 1, 2, 3, 5% single-digit yield, it's a non-starter. You can't really go very far with such a low yield. Now contrast that with biology. Biologists don't apparently care about yield. That's bizarre to me, but of course Sidney and Richard knew this to be true because you only need a tiny amount of a biological compound because of its ability to replicate or be amplified. So the idea was, what if you could amplify the information of a synthetic sequence of reactions by combining the informational power of biological molecules? So chemistry has no information in it, simply diversity, but biology has a potential to encode actual information. So the first proof of concept that this could work was by Kim Janda, who was with Richard, and he teamed up with Sidney. They published this JAX paper together with Richard, also published a patent that laid the groundwork for actually achieving DNA-encoded libraries in the lab. But look at the date on that, 1993. It would be still many years before people would fully appreciate the ramifications of these findings. So let me take you back to the beginning. Go through a history of how is it that people actually discover a drug. Now, the standard way, the conventional way of discovering a drug is you do a ligand screening and you make a large library, let's say combinatorial library, of different building blocks. And those building blocks are screened against, let's say, an enzyme of interest. And from there, you end up with a compound that might be an interesting hit, and that hit then becomes a lead, it's developed further, and then finally becomes a drug. But the process is slow because the numbers are low. Now let's compare that with a biological technique that Richard really was one of the key pioneers in, along with my brother, Carlos Barbas. So Carlos, of course, many of you know, was taken from us too early. I'm actually wearing a shirt that he gave me, and hope he's here too with us, watching. Anyway, he and Dennis Burton and their team with Richard worked on a process called phage display. What is phage display? If you don't know anything about biology, let me try to explain it to you. A phage display, it's short for a bacteriophage, which is basically a small virion molecule, and it displays a protein. And so what happens is you hijack these little virion particles to make tons and tons of different proteins that are all encoded with the little genetic material that is hidden inside the actual phage. So the phage contains the genotype, and the protein is the phenotype. So the information is stored in that little phage particle, allowing you to express and make millions, billions, 10 to the 10th different constructs. You then screen it across your enzyme, let's say, or target of interest. Some of them grab on like transparent, like tape, very strongly. You wash away all the non-binders that are not very sticky, and then the sticky one is identified. There may be only one molecule from 10 to the 10th. That's a large number to get your head around. Imagine there's one single molecule, but that's all you need because you can let biology do the rest for you. You just take that piece of DNA from the bacteriophage, use PCR, amplify, and the amplification tells you that one needle in the haystack exactly what it is. So now you see where we're going with DNA-encoded libraries. We're going to use the logic of phage display applied to organic chemistry. Now here is a typical DEL molecule. The colors on the DNA strand signify the synthetic steps that the individual molecule has been exposed to. So this is a way of encoding into the tag the exact synthetic steps that have happened. So you do your synthetic sequence on a particular molecule. You then mix them all together. Each time you do a synthetic sequence, you barcode. You add a little piece of DNA to say, that's what I did to you. You do it again in a process known as split and pool, which expands the numbers possible of compounds dramatically. You keep doing this for a sequence of events, and you end up with millions, if not billions, of compounds. Not in separate wells, in one tiny Eppendorf. So in a tiny test tube, you can hold billions of compounds. How do you screen them? The same as phage display. So your little tagged molecules, all in a well, get incubated on the surface of a target. Some of them stick, some of them do not. There may only be one compound. Who cares about the yield? The yield is irrelevant because all you have to do is get the result, amplify your answer, and then you know the synthetic sequence that you need to recapitulate in a flask. It's really brilliant. It's brilliant, but there's a big but. And Richard knew there would be a big but at the beginning. He knew that the skeptics would say, yes, that's all great and it looks awesome on paper, but this is never going to work. It's never going to work for a simple reason, that chemistry is not compatible. Organic chemistry is not compatible with the crazy rigors of doing chemistry on DNA. He recognized that, but he said in his usual fashion, the chemists will figure it out. And they did. Now fast forward a few years, the repertoire and toolbox of what an organic chemist could do on DNA, it did evolve, but it was still rather sparse because the only types of reactions one could do were things like condensations, amide bond formations, some reductive aminations on a good day, some paracyclic reactions, but the types of reactions you could do really had to be the most robust reactions of all time because of the difficulties of doing chemistry in the presence of DNA in aqueous media, and I'll talk about that in a moment, which gets me to my next lesson. How do we get into this? Well, it really comes to this nexus of academic and industrial research. I remember there was a day, even when I was a graduate student, there was within the community this pervasive idea that somehow collaborating with industry was dirty, that it made your work too applied or strange. Why would you collaborate with industry? For us, for the Research Institute, as Pete showed you, from a funding perspective, it was a complete game changer. And it set the stage for now something every school aspires to have in academic industry collaboration. Our group was primarily federally funded until 2010, when this company in Denmark called Leo Pharma came to me and said, we'd like to do a synthesis of a molecule. We'll fund 5 postdocs in your lab. Game changer. But the Office of Technology Department at the time was saying, no way, we can't do this because the terms they want, ownership of all the IP, it's too onerous. But if it wasn't funded, my lab would have closed down because I had no more money. So I called Richard and explained to him the situation, and it was kind of like my offer back in 2002. It was near instantaneous, like, yes, that's foolish. Of course, we'll make it happen. And that was the end of it. It happened. I think that written into the bylaws of Scripps, and maybe it might still be there, is ultimately ask for forgiveness, not permission. And Richard operated that way to the benefit of the faculty. And because of him, now 70% of the lab's funding is with industry. And our collaboration working on DNA-encoded libraries was facilitated by this very fact. So Pete and Richard actually orchestrated a brilliant collaboration with Pfizer that brought in an enormous amount of money to the Institute and funded 5 or 6 different teams to work on the problem of how do we get next-generation chemistry to work on DNA. Multiple groups were involved. Now, from an organic chemist perspective, when Richard first described to me what was going on with DEL, and I was reading the papers and learning about it, I realized that DEL is actually a nightmare, especially if you want to achieve a chemistry like this. The Pfizer people said, hey, Phil, it'd be great if you could make sp2, sp3 linkages that are architecturally rich. These are really hard transformations to do in an organic solvent with a regular small molecule, and they wanted to do it in the context of a large piece of DNA under the most rigorous conditions. It is a nightmare. A nightmare because it is really like doing chemistry in a toilet bowl. You have concentration levels that are millimolar, so the molecules never see each other. You have to use huge excess of reagent. None of your organic compounds dissolve very well in aqueous media. And not only that, all of the chemistry you do has to not touch all the functional groups on DNA. It's like ridiculous. And at the beginning of this, I thought, well, I sort of operated like Richard. I thought, we're not going to be able to do this, but they're giving us a lot of money, so let's see what happens. The key team behind this, and the reason we succeeded at the outset, was because of Richard's credo of when you don't know what's going on and how to do something, you should collaborate. So we realized quickly that mechanistically we would have no idea of how to control this problem of high dilution, getting the kinetics right. So we collaborated with the best mechanistic kineticist person on the planet, who Richard hired, of course, Donna, who's now the chair of the department, along with her postdoc, Helena, and 3 super brave postdocs in the lab who are now off on their own independent careers, but they were really brave to work on this problem of doing chemistry on the nanomole scale with ridiculous levels of chemoselectivity. And here's what Donna taught us. Donna taught us a rubric of how to take an organic reaction and systematically understand its kinetics in a normal reaction setting, translate that to the wacky world of DNA-encoded libraries. How do you take a reaction that takes place in the normal organic phase, normal concentration, no chemoselectivity problems, then go to the aqueous, doing chemistry in the ocean, basically, and not touching any of the creatures there. So with all of the learnings from that collaboration, we were able to actually do this radical reaction, which takes the starting material we call a redox active ester, which is a derivative of a carboxylic acid, that is actually just basically like an activated ester used for amide bond formation. Turn that into a radical, which would then intercept this Michael acceptor in a what's known as a Giese-type reaction. It took place in aqueous media, nanomole scale. Yields were almost quantitative in some cases, completely DNA compatible, because it worked because it was a radical pathway. And so here's an example of a normal reaction, we could do this reaction on a 1 mole scale, or we could go all the way down to a tiny nanomole scale. But Pfizer, although they were doing backflips after this, and I showed Richard and he was super proud, it was never enough for them. They said, that's good, now we're done with that, but what we really want is the aryl-alkyl bond. If you could do this transformation, that would be really great. Mechanistically, I realize not everybody here is a chemist, so I'll try to draw you through this. Mechanistically, you need a reducing agent, so you need to take that redox active ester and coax it to lose carbon dioxide and leave in its wake a very highly reactive species we call a radical. That radical then needs to react with a low-valent metal catalyst like nickel, and then a reductive elimination takes place, which is basically like the nickel is like a composer and tells these two pieces, hey, you two, come together. And it needs to do all of this in the presence of DNA. It needs to do all of this in super high dilution, multi-component coupling reaction in high dilution, and it needs to do it in water. So we tried this for about 8 months, and not surprisingly, after hundreds and hundreds of reactions, we couldn't get anything. And it was kind of a demoralizing period. Richard would be calling, how's it going? It's not so well, actually. And so we were embarrassed because Pfizer was breathing down our neck, and also the phone calls from Richard wondering how we were doing on this. So the fundamental problem is, like I've said before, how do you adapt these reactions to work in water? But I think the stroke of brilliance came from another colleague later who said, you're thinking about it all wrong. Instead of doing your reaction in water, what you need to do is bring your DNA into organic solvent. We had not thought of that. So another Richard Lerner hire, Phil Dawson, who's now the dean of the graduate program, a few years ago was working on functionalizing peptides and doing so in unconventional ways, and he realized that the way to solve the problem of insoluble peptides is to absorb them temporarily onto a solid surface through taking advantage of multivalent kinetics. That is to say, if you have a lot of sticky spots on your resin, the DNA, or the peptide in this case, has also a lot of compatible sticky spots, and so it will stay on the resin. The small molecules, though, will have much more of an off pathway, so they won't stick. So only the large molecule will stick. It worked really well for peptides. Has it ever been done for DNA? Actually, it had been. There was one precedent in the literature from Harbury. The problem with this precedent is that it only worked for a very boring reaction like a amide bond formation. Because the backbone of the solid support was a sugar, you would also functionalize a sugar. And you also had to do this in a basic solution. So it, again, didn't really solve the problem. So Phil and his team of one person, Dylan Flood, his student, solved this problem in a definitive way by coming up with a completely different type of resin, a strong anion exchanger that was completely inert and had an incredible magnetism towards oligonucleotides, specifically DNA. Super durable, cheap and commercially available. That's one hallmark of Dawson's chemistry, is just it works, and it's always super simple, very pragmatic. It's like a Scripps really logo of what people do here. So here is the proof of concept of how you could take, from Phil's lab in this patent, you could take a piece of oligonucleotide, you could bind it to this resin, you could wash the resin, and then you could either do chemistry on it, or in the case shown in the slide, simply elute it by changing the pH of your buffer solution. So it would absorb onto the surface, and then when you tell it to, gracefully depart. The brilliance of this now is that you can use these resins in organic media. So here's the dream. Can we use this RAS technology, reversible absorption onto solid support, to get the DEL out of water? Put these little pellets, this little resin, in organic solvent, do everything in high concentration, not have to worry anymore about DNA because you've removed and obliterated all the sensitive functional groups, because now it's just sitting on the surface. So here's the team. Phil, of course. We also were lucky to work with another young recruit. He was hired by both Pete and Richard, Ryan Shenvey. Jason Chen, who runs our automated synthesis and purification facility, and then the team shown here. A lot of people weren't pictured as a large team with Pfizer as well. And so we set out to now realize Lerner's DEL dream, once and for all. So in the first proof of concept, we did a boring reaction, that is just to do a amide bond formation. Look how clean it is. The really startling thing is now you can do these reactions in the organic solvents that we know and love. Dichloromethane, toluene, DMSO. those are like, the paints of organic chemistry. We love to use those solvents. No more water. We hate water. So finally, we could do these in normal conditions. And then the key question was, where the rubber hits the road, could we do this dream reaction that Pfizer had been begging us to do for years at this point, and which we had failed miserably? So here's the result. We could take this large oligonucleotide that has appended a very tiny aryl iodide, which is a unit that is primed for cross-coupling. As you can see in blue, which represents the sort of aqueous conditions, we get nothing, of course. The DNA is completely trampled over and destroyed. When we use the Dawson technique of absorbing it onto a surface, combined with the Shenvi reducing agent, we now can get in the 80s for a transformation that I think would be really admirable even just without the DNA, just by itself. So here's a typical chromatogram, super clean. The DNA is recovered, not destroyed at all. Take a look at that scope. I realize that many of you are not chemists, so you won't appreciate it, but all of the DNA is notated on the left. If this table didn't have any DNA, it would still be a really good paper for an organic chemist to see you could do this transformation. But now you're doing it with this ridiculous piece of DNA hanging off of the side of it. So this technology was a real game changer for us and ultimately for Pfizer and their CROs that were doing DNA-encoded libraries. We then decided to up the bar and wonder how far could we take this? Historically, DNA is redox active, and in principle, it should be easily obliterated if you expose it to an electrochemical apparatus. It should be electrocuted, really. So if it really is being protected, could you start doing exotic electrochemical reactions on the surface of these particles? Would it work? Which gets me to another lesson, something that wasn't mentioned, and I think many of us have the same recollection. On running a research group, I would often get these calls from Richard. It happened on a couple of occasions over the years when we published something that was particularly controversial or different. He would call me up and he'd say, Phil, I heard about this thing you're doing now, and I got some calls from some senior members of the community. They really don't like it. And I would stay quiet and wait for what I didn't know what was coming, and he would say, you should keep doing it. He loved it. I think many people who care about what other people thought, they might have said, oh, be careful, maybe tone that down a little bit, don't do that so much. And one of those things was electrochemistry. And we got into it, and people were like, oh, we've seen videos online, this is a little weird. Tell this kid to stop that. But we just followed our instincts. And sure enough, a decade later now, it also makes the cover of CNE News. People seem to think it was okay. So does electrochemistry work with DNA? It does. Thanks to Phil Dawson's technology, we can now do amination, electrochemical amination with a ridiculously good scope. We could even go to boring reactions, like reductive amination. That's like one of the first reactions you learn as an organic chemist. But as it turned out, the Pfizer people confided in us that even this simple little reaction that everybody learns as a first-year student didn't work very well. The scope was terrible. So we could, again, use this RAS technology to change the game and allow for even simple reductive amination on substrates that they knew didn't work very well to now work really well. Again, the scope, really good for branch substrates, the type of substrates that Pfizer said to us, these don't work, and now they did. So what does the workflow look like in this next generation DNA-encoded library synthesis apparatus? And we did a mock library to show you how it works. It was a 4-step synthesis of which 3 of the steps made key skeletal bonds. And all of the bonds we made were ones that were either very difficult in any other way or conventional reactions but didn't work on DEL. The first was a decarboxylative cross-coupling, then an FMOC deprotection, followed by reductive amination on a branched ketone that we know doesn't work well, and then finally an electrochemical amination, all on DNA. You can see the workflow is on the left. You basically absorb your DNA to the surface, you get rid of the water, dissolve it in organic, do your reaction, then remove the organic, take the DNA off the support, put your barcode on, and then repeat the cycle over and over again. And after that, we got 9% of that product over 4 steps, which is a fantastic yield for DNA. As I mentioned, you really only need trace quantities because guess what? We can amplify. So how does it work? There's a sort of overview of it. Everything in yellow, those are the steps which you previously couldn't do before. The things that required organic media now can be brought into this RAS setting to enable these types of reactions, to make libraries of 10 million in a tube with chemical diversity that would have been unheard of. So over the years, we continued to publish on this. We also published a method with Dawson's group on doing electrochemical reactions to make carbon-sulfur bonds, regular reactions to make carbon-phosphorus bonds, carbon heteroatom bonds, the ones that Pfizer said, these are the ones we need, folks, and we have no way to do it. If you want a deep dive into what is DEL, where did it come from, where is it used now, and the modern variations of it, we wrote a review in Udi's journal, an Israeli Journal of Chemistry, that Richard liked quite a bit, that goes through it. If you're one of the scientists in the audience who would like to learn more about Richard's legacy in this area, please feel free to take a look at that. So finally, one further lesson that Richard taught all of us, that Pete alluded to a little bit, is this one. You know, when I was a student, there was always this perception that scientists should be like Nikola Tesla, die alone with a pigeon as your wife, bankrupt, with a test tube somewhere, right? It was always this vision of the poor scientists with cobwebs everywhere, and they died there in their lab. Richard was not like that. He encouraged people, whenever you talked to him, he said, oh, that's great. Have you thought about making a company based on that? Or did you patent it? You could make a company based on that. Like, there was no shame in having pragmatic and commercial applications of your research. Why should there be? It's ridiculous. And so he really broke that barrier down, as you could see from the Porsche and Ferrari pictures. So, and I should mention, as a corollary to that, that Phil Dawson and I just co-founded this company about a year ago, ELSI, and its core technology is building on the learnings of what Richard Lerner taught all of us. Okay, so the last few years that we had with Richard, it was great to visit him. He was full of wit, and although he did talk a little bit slower, he was 100% there and still completely focused. Here's a picture that from when Jen and I visited him about a year and a half ago. And we even visited him about a week before his passing. And what struck me about those meetings with him was his obsession with this machine. You might think that's weird. He was obsessed with this machine. He asked multiple times in the last meeting, when is this machine going to get set up? When is the department going to be able to use it? He used his connections to allow for the purchase of these two enabling LC-MS machines for the automated synthesis facility that would benefit the entire chemistry department. And he was so focused on how he could make our lives easier. He had called me up a year before and he said, I want to do something for the chemistry department. What would be a game changer? And then every subsequent meeting we had with him, he just wanted to know, when was this going to be installed and how could it make your life easier? He was full of love, and the way he expressed his love was by trying to help the people he loved do the science that they loved. Even the automated synthesis facility, he worked hand in hand with Pete to be able to raise the money from the Picower Foundation that now funds this department that has, I think, really revolutionized, along with Jason, how we do chemistry in the chemistry department. Every student and postdoc benefits from this. It's generated, I don't know, 70 different papers. I mean, it's an amazing facility, and Richard's hands are all over that. In closing, here is a picture of Richard and I at his favorite restaurant, Red Tracton, down the street in Del Mar. And during that dinner, I remember he was telling me all about DNA-encoded libraries. I was putting this talk together, and my 7-year-old walked in, and he said, Daddy, is that your father? And it struck me because he was more than that. And I am grateful that I was able to know him, and I'm also really grateful that I was able to tell him that several times before he passed. So with that, thank you very much.
We can have some Q&A for Phil if we have some questions. Maybe I have one. I mean,
Sure, Jamie.
so, I mean, there's well-known organic chemistry to actually synthesize nucleic acids on solid supports. And so in that mode, you could sort of get around some of the problems associated with being aqueous. So did you guys ever discuss that sort of parallel synthesis with conventional DNA and other chemistry? And
That's a great question. Here's the issue is that I think it's really cool to talk about this, because if you do oligosynthesis on a solid phase and you swell the resin and you start trying to do chemistry, the functional groups on the oligonucleotide are still exposed to the reagents. The brilliance of Dawson's strategy is that it takes a piece of DNA and it throws a blanket over the whole thing. So even though the reagents are there, they can't see the DNA anymore. So that's what, and the best part of it is there's no extra step of a covalent bond needing to be broken. So for Phil's strategy, all you do is change the pH, and the thing says, okay, talk to you, see you later. The blanket comes off, and the DNA departs gracefully. There's no covalent bond that needs to be broken. So that's why we couldn't do it with this. Yeah, it's a great question.
Makes sense.
Yeah.
Other questions? Here's I can't see who that's.
Scott.
Scott.
So, yeah, very fascinating. How far along do you think you are in the chemistry? So I'm looking at the publication dates on those, and they seem like they're quite recent. So of the chemistry that really needs to get done, how far do you think you are, and where do you think that this is going?
Well, that's a great question. I think the funding from Pfizer had ended, and we achieved the mission. We gave them the deliverables they wanted, and so I think they licensed Phil's patent. They are using the technology out not only within Pfizer but also at a company they work with called HitGen. It's been reproduced there. So from our standpoint, our job from an academic standpoint is done. And now it's in the hands of industry to, I think with the RAS technology, you can make enough diversity that you need to get the hit. All you need for Dell is not the final drug candidate. That was never Richard's vision, to give a final drug. He just wants to accelerate the process to get you that lead. And then conventional medicinal chemists with a 1 micromolar hit and some good structural information can turn that into a nanomolar inhibitor in a few months. Hey, MG. Great to see you, brother.
Last question.
So I'm going to ask you a Richard question. So Richard deposited in my brain several ideas which I just wasn't good enough to bring to fruition. Is there anything in your mental suitcase that he deposited there that you're still working on, or that you wish that could come to a state where you'd be willing to talk about?
It's a great question. And actually, we have a paper, Nikki knows well, we have a paper under review now where it actually has to do with some CAR-T chemistry. And we also had a paper we worked on with flavin chemistry. And so there are some ideas I've jotted down over the years. Some have actually resulted in publication, but the one that was sort of the most impactful for us was the story I gave today, just because the combination of what we were doing and what he sort of wanted to achieve, we kind of viewed this as a gift to him to finally make that dream of his, be something, and that could actually. So the whole Scripps Institute, Phil and Jing Kwan was involved in this, and Ryan Chenvey, even Barry Sharpless for a time, Pete, we're all involved in this Pfizer thing to try to fix Dell, and we're happy that it finally worked. But you're right, he's got a whole slew of amazing nuggets of wisdom that can be converted into companies and science papers. Yeah.
Okay, let's thank Phil again. Great job. Okay, our next speaker is Professor Raymond Dweck. He's a professor at the Department of Biochemistry at Oxford University. I think it's safe to say that Raymond is one of the founding fathers of the field of glycobiology. And perhaps not surprising, he founded the Oxford Glycosciences Institute, and he's a founder of Oxford Glycosciences. He has served as the director of United Therapeutics, and they've developed a number of FDA-approved drugs. I think one of the things that I'd like to lift up about Raymond was that he helped along with Ian Wilson and Richard, to set up the joint Scripps Oxford PhD program, which has been an exceptional asset, bringing some of the best scientists in the world to do their graduate studies at both Oxford and Scripps. But I think that it's significant. I am told that that's the first joint program that Oxford had ever set up in its 800-year history, and this was really the brainchild of the three of them. So it's been a pleasure to work with you, Raymond, and please come to the stage. We'd love to hear your talk.
Thanks, Amy. Well, good afternoon. It's, for me, it's a very emotional time. The passing of Richard as a very close friend was a severe blow to me, even though we knew that it was almost inevitable. Because as Richard would appreciate as a chemist as well, the breaking of bonds is never easy. So, in the history of Oxford University, as you just heard Jamie say, 850 years history, Richard has played a very significant role. And as the Skaggs was mentioned, let me start with that. In 2002, the Skaggs family visited Oxford, and they wanted to have an interaction with Oxford and some studentships. Richard thought it would be better done through Scripps, and of course we in Oxford agreed with him. Let them do all the work, we said. And then all of a sudden, Richard arrives, and he speaks to our Vice-Chancellor, John Hood. And John Hood is unbelievably supportive of the idea. And Richard comes up with an idea that no one's ever had. We will have a 5-year program. They'll get a PhD from Scripps and a DPhil from Oxford. Impossible, everybody said. All the administrators, and there are more administrators than you can think in Oxford. They all say it's impossible. Do you know that Oxford is run by an Act of Parliament? It has to go to the Queen to sign a Privy Council. So Richard said, well, you and I have agreed. I'm sure we can get the Queen on board. It's your job, he said. So Richard shook hands with the Vice-Chancellor. They went out, and a year later, it was signed by the Privy Council, unprecedented. Princeton and Harvard immediately followed, and they were turned down by Oxford, by the way. We offered Princeton an MSc program, but nothing like this. This is a unique program that goes on in Oxford. But it's worked really in Oxford because of the dedication of Ian Wilson, who's been a mentor to most of the students involved in that. So it is a great program, and it's a flagship really for Oxford as well. It attracts very good people. As a result of that, Richard was asked to join the Court of Benefactors. And here you see him with Lord Patten. And the room in the middle here, that was where Charles II had his parliament. And it was in that parliament, in that place, 2 years later, you saw this picture with Pete's presentation, that Richard was given an honorary degree acknowledging his work, pioneering work on catalytic antibodies. And those who know Richard well will note the Patek watch on his lift, and the pen, which is a Visconti pen, was another of Richard's hobbies of collecting pens. His co-honorands those days, including Daniel Barenboim, the musician, Lord May, Jimmy Carter, who is not named here. Then there was the Lady Hale of Richmond, who took on the government recently in the UK, Clive Granger, the Nobel economist, Minushkin, the producer, and Rao, the chemist. And the degree ceremony took place in this building over here, which is the Sheldonian Theatre in Oxford, designed by Wren, Christopher Wren, who built St. Paul's Cathedral in London. But he designed this in 1664. And it was in that building 2 years later that we had another ceremony celebrating the 200 years birth of Darwin. I was the president of the Institute of Biology, now the Royal Society of Biology, and I decided to recreate the Darwin debate. This was a debate between Huxley and Wilberforce on Darwin's creation theory, and it took place in the Natural History Museum in Oxford. And so I had the Bishop of Oxford and Richard Dawkins debate with each other, and I had Jeremy Paxman, the television presenter, moderate. But to give the bishop a chance, I had Haydn's Creation put on in the Sheldonian Theatre, and then afterwards we had the ceremony here, and you can see here they all were awarded membership of the Institute. Richard Dawkins, Gunther Blober, we know, and Barry Bloomberg. And on the left is the public orator from Oxford University who made sure that all the proceedings were carried out correctly and in Latin. Richard Lerner's love of chemistry was something that he always brought to Oxford. It was constantly talking about medicinal chemistry. And when Andy Hamilton, who had been at Princeton, became the Vice Chancellor of Oxford, he was very, very keen to try and bring more medicinal chemistry into the biochemistry department. And he encouraged me in the Institute in 2014 to set up a Richard Lerner lab on medical chemistry. History was made another 2 years later when, following a donation from the Lerner family and Marjorie Fink, we set up the Lerner Medical Fellowships, Lerner-Fink Fellowships. And the first one was J.L. Kiepis from here, who'd worked with J.C. Nicolau, had been a Skaggs Scholar, was the first Link Fellow, and is now in University College London as a professor. And they worked under the leadership of Nicole Zitzman, Professor of Virology. One of the other functions that Richard helped me with was he was on the board of the biochemistry department, which, as you know, has 1,000 or 2,000 people in it now. And I had 7 buildings, and I was trying to unite them all into one building. So I had this very distinguished panel that actually helped me set up this, and now the building has just been opened in 2021. It's called the Dorothy Crowfoot Hodgkin Building, and in there, biochemistry is all now united, and the first Lerner-Fink Fellowship in that building is Julie Brunn, some of whose work I'm going to describe in a moment. And if you go into the side of this building here, the west entrance, where most of the students sit and eat, if you go into the inside there, there's a plaque there which recognizes the support from Scripps and really testifies to the importance of the Skaggs Oxford DPhil program. Some memories here of, is this a pointer? Yeah? Yeah, some memories here. So here's Richard in Oxford at some function. You can see Ian Wilson there with Marjorie Fink, Dennis and Carol Burton there with my wife Sandra. And in the background is Martine Rothblatt, the CEO of United Therapeutics, who came here to meet with Richard and was very, very impressed with Richard. And we have a glioblastoma antibody there, which is used in pediatric cancer, very successful. And because the original cell line was done here, Martine offered Scripps a royalty quite voluntarily. Then you can see Alan Murphy Lerner, who this is taken when they came to Oxford. Here's a young Richard Lerner in the year 2000 with Nikki. And I want to say that in all of everything we hear about Richard, you have to understand the rock that Nikki has been for him in all of these years. Not only telling him when he's gone too far, but just being there. And I always appreciated that. Here you've got the Frosts, and there you've got Hugh Rosen and Deborah Rosen, also who were Oxford educated, with Richard. So I traveled the world with Richard when I think about it. Over here we had San Francisco. Sorry, let me go back. San Francisco, when we went to meet secretly with Jeb Bush to talk about setting up the Florida campus, Utah with the Skaggs family, and then places in Florida, of course, New York, and it went on other places in the United States. And you can see right across the globe over there. And I thought I'd just highlight some of the ones that I remember. In 2003, Richard got an honorary degree in Ben Gurion University of the Negev. And you can see here, he was clearly enjoying himself very much at that meeting. And he's on a panel here. There's Phil Needleman there. There's and you can see me, I'm sitting in the middle there, covering my head as Richard's making some outrageous comments. And I'm trying to think how I can actually finesse this without offending everybody. And there's Richard sort of smiling angelically in the corner, knowing full well that he's landed me in a position. And then in 2009, he was honored by the University College of Dublin with an honorary doctorate. But really what Richard liked was he got the Ulysses Medal, which is named after James Joyce, who's widely regarded as the father of the modern novel and the most seminal author of the 20th century. So I put Richard's books here that Pete talked about. And I think Richard was always I always said to Richard at the time, we should mention your books. And Richard said to me, under no circumstances mention the book. If you do, I'll have to leave. And then, of course, in 2009, we go off to Korea, the Scripps Career Antibody Institute. I did a lot of traveling with Richard and Sydney. Sydney spent a lot of time also in Oxford. And in Romania, in 2010, Richard and Barry Bloomberg were received by the Academy. And Richard actually had a nice time there telling people in Romania about what science could be like and should be like in the future. In 2012, we accompanied Richard as guest of the Queen of Spain to the Prince of Asturias Awards. And it says something that the Vice Chancellor of Oxford, Andy Hamilton, now he's the president of New York State University, he actually came as well to support Richard. So strong was his feeling and support and admiration for Richard. And there you can see the well-known gang over there. You've got Al and Muffy Lerner and their daughter. And then, of course, you've got Ian Wilson, who was amazingly present at most of these things that I went with Richard on. This picture of Greg Winter and Richard, it took a whole morning to produce that picture. You know, it wasn't just one take. Finally, they got the picture that they thought was good. In 2012, Richard helped establish the Shanghai Tech aspect of the immunochemical studies. And here he set up a governing body board. And a measure of the commitment was that Nikki started to learn Mandarin. And I know that Nikki and Richard, who's listening, would like me to thank Guang Yang, who did so much to make life so easy for all of us. And we heard from both Phil and Pete about the DNA-encoded libraries. It was Richard's idea in 2018 to make this available to academics. And that he set up here in the Scripps, a board of which many Scripps members were on it. But again, it was another aspect of Richard wanting to help people to leverage new technology. My last interactions with Richard concerned the neutralizing antibodies to SARS from a library obtained in 1999. And they found in that 3 antibodies that co-reacted, that cross-reacted or co-reacted with the spike protein. And we must have had a huge number of phone calls on this. And the phone would suddenly go on a Sunday afternoon, and there would be Richard. He didn't really mind about the time difference. He didn't seem to bother him if I was in the middle of dinner or whatever. And he would just talk for an hour. And we talked, and this was, I remember, lots and lots of talks on this. And I remember that in 20 years ago, he gave this lecture in Oxford. Antibodies yesterday, antibodies today, and antibodies tomorrow. And there you see a picture, again, a younger Richard. But I first met Richard in 1981. Dennis Burton had just been working in the lab and had solved the C1q binding site on the antibody molecule. And there was a CBER meeting, which was a closed meeting. It was limited to about 20 people. But Rodney Porter had arranged that 2 extra people should go there. One was Richard, and the other was me. And that's where we first met. And I remember it was a good meeting, and we remained very firm friends for a long time afterwards. And I told Richard at the time that my interest was really switching from antibody binding sites and the inflammation, the defense aspects of the antibody, to the role of sugars. And 3 years later, we developed the technology to do that. On an antibody molecule, we found, didn't just have 1 or 2 sugars on, but had the whole collection, and here, 36 structures. Now, I remember phoning Richard up, and he loved the idea, because for him, the idea was you've now got diversity at both ends of the molecule. And so I came up with the concept that we can pair these up, and maybe we could call these glycoforms. And these glycoforms may have different functions. And Richard liked that idea because, of course, he wanted diversity there too. And of course, one of the first aspects we found that these sugars on the antibody molecule, and here's a fingerprint, if you like, or a sugar print from a normal person, and you can see here you've got lots and lots of different sugars. I want to point out that over here, you can ones that are G2, that means they've got 2 galactoses on. Look at the ones at the end. It's not showing up on here. G1, they've just got 1 at the end, and G0, they have none. You can see here, there's nothing here. So this was a rheumatoid patient. This was a normal patient. But then when we looked at the fingerprint for somebody with rheumatoid arthritis, it was very different. And you can see here that those with no galactoses on had become much larger. Now, this decreased with anti-TNF therapy. And again, that really pleased Richard because it showed that we could alter the course of the disease. But altering the course of the disease, really, the idea of that really came from Dorothy Hodgkin. One of the things I had to do as a young man was to help carry Dorothy's bags when she went to dinners in the Royal Society. I was a young fellow. And during that, she told me her entire life story. And she told me that she got arthritis very badly after her first child. But she had many miscarriages. And she always knew when the miscarriage was going to happen because the arthritis would come back. And of course, we now know that pregnancy is a very immune-privileged time. And before pregnancy, this is a rheumatoid profile. During pregnancy, they go into remission, and postpartum, it comes back. So that led to the idea of looking at what was the function of these sugars on the antibody itself. And here, in this particular instance, these sugars that had no galactose on bound to a protein here, the mannose binding protein or the mannose binding lectin, and that triggered the activation of complement. So you could say these were inflammatory glycoforms. And we then found, of course, that did another active story, not with Humira, but with infliximab, the same thing really, showed that, again, the anti-TNF therapy could cure the progression of the disease. We still didn't know what the mechanism was, but Dennis Burton came up with an intriguing possibility that the IgG secreting cells may have receptors for factors such as inflammatory cytokines, which may modulate the glycosylation machinery. That's still to be worked out. So we had here pro-inflammatory forms, which I've just talked about, intravenous immunoglobulin, which we know is anti-inflammatory, and that ends in sialic acid. And if we look at the antibodies produced against, say, COVID or HIV or enveloped viruses in general, then they are defucosylated. And that we can explain that. It allows the Fc gamma-3A receptor to bind more tightly and activate cellular responses. So that's an idea of these different glycoforms having different activities. Well, that was really what I imagined at the beginning. So when I defined the word glycobiology in 1988, I speculated that this is what we might find. And I was delighted when Oxford English Dictionary approached me. One day, a lady knocks on the door, and she says, I need to interview you. I say, what do you do? He says, I do the letter G in the Oxford English Dictionary. So I immediately said, and how many of you are there? She said, oh, well, there's only 20, because we double up, you see, on two words. So she said, we want to put it, the glycobiology word, the definition, in the Oxford English Dictionary. And it was a great big celebration. I remember the Vice-Chancellor ringing me up and said, this is the greatest honor you'll ever get. Forget about prizes, Royal Society. This is it. You've got it in. And then I remember in 2010 being here when Nikki and Richard came back from Del Mar. They'd just seen this film, Extraordinary Measures, which is on a storage disease. And there's he turns to the waitress at the time and says, "What's the matter, Sal? Have you forgotten your glycobiology?" So the idea that I had for glycobiology, the idea was to put sugars in the context of the molecules to which they're attached. And you can see here on the SARS spike glycoprotein that once you put the sugars on, you've really changed the nature of what is presented to the immune system. And you'll see later that these sugars will give you a fingerprint of what that virus is doing, where it's been in the cell, and also sugars are quite important in how the virus folds. So Julie Brunn, who's the Lerner Fink Fellow, took the original Wuhan, or the strain. Actually, it was a strain which was again cultivated in England in February. And she looked at the glycosylation of it. And she looked at every single site. This is an absolute tour de force. And she found that some sites were less processed, but she also found that there was a mannose 6-phosphate sugar on some. And that immediately made us realize that that tells us that the virus is exiting here. Whoops, sorry. It tells us that the virus is exiting through the lysosome over here. So if we look at that, sorry, I'm just gone ahead here. Let me go back. So we've got here the virus goes into the cell. It assembles in the cell. It comes out and gets glycosylated through the various departments, and then it exits via the lysosome. And how do we know that? We know that a particular residue, gluconag phosphate, is put on first in the cis compartment, and then is taken off in the last compartment, the trans compartment, and that targets it to the lysosome. So the glycan signatures have told us which way the virus is getting out of the cell. And then she made the non-stabilized trimer, which was the Shadox vaccine that Oxford was putting out. And what she found was that when she isolated it and looked at the vaccine antigen, the majority of overexpressed protein of the S1 was actually in the serum and not attached to S2. So she, again, compared the two sites, and we could see, just looking at it, that they were different. And then we could say here, this particular instance, that the virus, it comes into the cell, assembles, goes out of the cell, and then starts to fall apart here because it's not stabilized. Now, I wrote to her right at the beginning about that to point out the work of Ian Wilson and Andrew Ward, which we'd learned from the HIV, on the importance of stabilizing the trimer. And both Pfizer and Moderna had stabilized their trimers. So this was, they took it sadly as a criticism, but I said that it's worked, it's been in 3 billion people, but if you do it again, don't make the same mistake. So that leads me to the idea of what we would need in a pandemic preparedness. Well, you've got immunomodulatory drugs, direct-acting antivirals, or host-targeting antivirals. And I favor very much the idea of host-targeting antivirals, but they remain the focus for the treatment of early disease. But host-targeting ones, we don't need to know what the virus is in detail. We're targeting the host, not the virus. There's a high barrier to genetic change. You're not going to change the host. So then you look at the world and look at the viral infections. They're a global issue. And look at all these alerts we had from 2003, avian flu, SARS, influenza, Zika, Ebola. And what did we learn? How ready were we? Well, we weren't. And if you look at COVID, an example of preparedness, here's COVID, 2019. Look at the first in red, the vulnerability levels. Nothing happens. You have an unmet medical need that I believe can only be targeted by a host-targeting antiviral, because you do not know what the next challenge is going to be. It has to be virus-independent. And so what do all these viruses have in common? Well, they need our glycosylation machinery. So that is the Achilles heel, I believe, of the virus. So if you look at how the sugars are involved in helping the virus achieve its three-dimensional folding, let's just look at some basic biology here. You have some sugars put on after it's growing on the ribosome. It has 3 glucoses, GGG at the bottom, and they're sequentially trimmed down until you've only got the one at the end. And that one needs to enter a cycle. But supposing you inhibit them there or there, then you can actually stop it going further on. So these are structural targets. And the molecule that I'm taking, I hope, at the moment to the clinic is an amino sugar, which I'll talk about later. Once you've got to the single method, the single molecule, then it comes in, it's held here by a molecule called calnexin, and other chaperones can work on it. If it's folded, follow the green arrow. It's correctly folded, the viral glycoproteins can assemble, and it's exported. If it's misfolded, it gets another chance. Another glucose is put on, and it goes on the red arrow all the way back and can go round and round. Now, you're targeting a host protein, so you have to know that you can actually do that safely. And so what happens if you inactivate this cycle of folding? Well, it turns out that this cycle of folding seems to be mainly for viruses. And so these experiments were done that suggested that these are my words, an Achilles heel for exploiting and designing antiviral therapies. And then there's another aspect I put in. This is technical, but it turns out there are 2 conformations of this molecule. One binds to calnexin and the other doesn't. So you can bypass the calnexin, and then if you bypass the calnexin and you've inhibited the pathway, well, there are enzymes that take off the sugars and put you back in the pathway. The endomannosidases is over there. So I'm reasonably convinced that we can not damage ourselves by targeting a host enzyme. So where did I get the ideas originally came? In the 1987 AIDS crisis. So Margaret Thatcher made a cabinet of 12 scientists, I was one of them, and we met in Sidney Brenner's rooms. I remember, and she came in and she said, Max Perutz and Raymond Dweck, you'll do the antivirals because we'll have a vaccine in a week. You know, we're British, we do things rather well. And I think Sidney had some very strong words to say to her, which I think delayed his knighthood. So Max and I went around the country trying to get people to work on viruses. It's incredibly how difficult it was at that time. And I looked for compounds, and naturally occurring compounds. And I went to Scripps one day with Max, and they were isolating molecules from plants. And they had an aqueous layer and a chloroform layer. And they always threw away the aqueous layer. So I said, can you send me the aqueous layer to Oxford? And we found in it, from the leaves of the mulberry tree, this amino sugar, which turned out to have antiviral properties. And you'll see later that we did work on it, but we had no idea at that stage what the mechanism was. It wasn't until 1993. But by this time, the MRC and Max and I had convinced Searle, Monsanto Searle had given me a large sum of money in Oxford, 100 million or something in those days, to develop the field of glycobiology. And we've convinced them that they should actually make the amino sugar, and they did. But I have to say, it was later when Chui Wei Wong actually made a better one-pot, 3-step synthesis that changed the whole field. So they found the antiviral drug did have some antiviral activity, but there was a side effect of osmotic diarrhea, and these were very compromised patients. So they stopped the trial, but I kept on studying it. And in my institute, and with the company I started, Oxford Glycosciences, we took it and developed a drug for Gaucher's disease, which is, again, targeting a host enzyme. And I have to say that 20 years later, it's been in the clinic, thousands of people have taken it, and there's not been a single serious adverse event reported. But it made available large amounts of the drug. And so we could continue our work with it. And one of the aspects, of course, you would expect, if it's really targeting a host enzyme, it should be blind to the mutations of the virus. And this is work done in Dennis Burton's lab here, where we looked at HIV-1, HIV-2, 31 isolates that were triple drug resistant, and we found that this amino sugar worked against them all. And then Barry Blumberg, who'd got the Nobel Prize for hepatitis, approached me and asked if he could join the institute in Oxford. And he had this idea now. He said, look, the 2 billion people still infected with Hep B. We need something. We need to think about antivirals. So we tried this in HPV, and of course you can see it seemed to work very, very well. So Barry was so convinced about this that when he had his official portrait painted for Balliol College, Oxford, it was under a mulberry tree, because he wanted to make the point that he thought these antivirals were something he ought to consider about. And it's that time that Nicole Zitzman joined the Institute, and I started to change the Institute towards more antiviral research. So with Barry and Tim Block and a few others and colleagues in the States, and Bud Tennant particularly, we started to look at the woodchuck model for hepatitis B. Notice we were developing more potent immunosugars, and we were just changing the side chain. And you can look at here at the top one, look at the glycan profile, and look at it 3 weeks later, there's hardly any difference. And yet many of those woodchucks were cured, if you like, from Hep B. But when we went to a clinical trial in humans, it was slightly toxic. So we modified it by putting an oxygen in it. And again, you can see here in dengue, both in vitro and in vivo, that it works. And Nicole Zitzman's group have recently co-crystallized it with glucosidase 1 and glucosidase 2. And so now we can make new compounds that, looking at the combining site and so on. But the question is, which target? Glucosidase 1, where you've got 3 glucoses, or glucosidase 2, where you're going to have just 1 left. And if you follow the green arrows, so the sugar is attached to the protein, the sugars are chopped off, and if it works, it's exported to the Golgi. If it doesn't work, follow the blue arrows, it gets another chance and goes round. And if it still doesn't work, it's terminally degraded, and it goes out of the ER-associated degradation. So, but that gives us a trick, because in this ER-associated degradation, free sugars, free oligosaccharides are formed from every stage. So we can analyze these, and we can look at the flux through there, and we work out that at low concentrations, you hit glucosidase 2, and at high concentrations, you can hit glucosidase 1. And you see it on this slide here. So here, the lower concentrations, you're hitting the blue one, you've got 1 glucose left. Hit the red one, high concentrations, and you've got 3 glucoses on there. Well, why is that important? Well, it turns out that there was a paper published in the New England Journal of Medicine where they found 2 children, 2 siblings, that had a deficiency of glucosidase 1. And they were resistant to viral infections. In fact, this is very rare, and in spite of the significant hypergammaglobulinemia, the children had no history of viral disease and were not able to generate an immune response to live viral vaccines. And the cells did not support replication of phylogenetic divergent species like HIV and influenza. And they conclude that these amino sugars like these should be used when there is for infections that pose a threat to rapid global spreading. And so attention was shifting to glucosidase 1. So this was the convention we were doing at the time, a clinical trial. Here's a mouse, give it dengue, 8 hours post-infection, 3 times a day, give it the amino sugar. And then we did this Richard experiment. There was a lot of stuff left over. And I remember talking to Richard at the time, and I said, we're going to do something silly. And of course, he loved it. I said, we're going to give a massive dose to the mouse. And I thought I'd get into trouble with, ethics and everything. They gave the mouse a large amount of the material, 10 times the single dose, one shot, and all the mice lived. So 10 days later, when they harvested, you can see that mice lived, it was just as effective as giving it 3 times a day for 10 days. And so I said here that it really began to make us think again, that the protection correlates with inhibition of the first enzyme. So it shifts the attention. Could that be the broad antiviral spectrum target that we could administer in a single dose? And just to show you it works against both influenza, we tried it on influenza H1N1 and H3N2. So a single step seems to be very, very protective. So we began to think about the possible mechanism of action, and this is speculation. So a cell with a large number of unfolded proteins will activate proteins in the so-called unfolded protein response to try and prevent the proteins accumulating. So we did some transcriptomics on this in dengue, and we found that the aminosugar, the drug, induces a robust unfolded protein response. So you've got one there. So now we look at the unfolded protein response. And what the cell wants to do, there's several mechanisms that it can bring the cell back into equilibrium. It can increase the protein chaperones. It can increase the rate of degradation, get rid of them, or decrease the protein production. But when all else fails, you get apoptosis. The cell dies. And so the theory that I'm working on at the moment with the Institute is that you have an unfolded protein response from the virus, and you have an unfolded protein response from the aminosugar. And when these are in the same cell, then you get apoptosis. So I think that's a lovely mechanism if it turns out to be true. And so now we have aminosugars as a broad-spectrum antiviral, and that's the latest. You see Omicron, it's really terrific against that too. So the aims at the moment is to prove that a host-targeting antiviral is active against humans, against current and future variants with a good safety profile when we use it in the high-dose regime. And I'm hoping that we can do a human phase 1 high-dose approach followed by a platform trial or a human challenge study, and also a single high dose for the treatment of influenza, which I think is bound to be coming now in the next year or so. So in future, we want to prevent epidemics from turning into pandemics. We want to stop the spread early with an already approved broad-spectrum antiviral. And I think that it needs a host-targeted antiviral, as direct-acting antivirals take too long to develop, and repurposed ones are not usually very active. So recently, I put these ideas together with John Bell and Mark Feldman and Nicole into an article in the Lancet, where I said we believe that the gravity of the pandemic urgently demands us to try the so-called off-the-beaten-track host-targeting antivirals rather than only the current acting antiviral approaches. And with a number of clinicians, I said the same message in a perspective in the PNAS, in which I said, to provide the readiness the world needs and deserves, we must learn from the past, but think differently in the future. And host-directed ones may be particularly important. And you see the window of missed opportunity that I've got there is in the first 6 months of when the virus starts to spread. So we've come a long way from glycobiology to antivirals. When Fred Sanger opened the Institute in 1991, we were doing all kinds of mainly sequencing, and we've now moved on. And it's all going to be incorporated into the new building under Nicole Zitzman. And I'm now making sure that all the research funds from glycobiology will be channeled into this antiviral approach. So on Richard, I could say on his science, traveler, there is no path. The path is made by walking, and that's Richard. His favorite quote, if at first the idea is not absurd, there's no hope for it. And on our friendship, think where man's glory most begins and ends, and say, my glory was I had such friends. May his memory be for a blessing. Thank you.
Thanks, Frank.
Okay.
Okay, thank you very much, Raymond. Our next speaker is Erling Norby, who is a world-renowned virologist. He's a professor at the Center for History and Science at the Royal Swedish Academy, and he served as the Secretary General for the Royal Swedish Academy for 6 years, and he was responsible for oversight of the Nobel Prizes in Chemistry and Physics. He serves on the board of the Nobel Foundation. He's currently the Vice Chair at the J. Craig Venter Institute. Welcome.
So thank you very much for that introduction. It's a great pleasure to be a part of this unique celebration, highlighting a life of really a unique individual that we, all of us here, have had the privilege to learn to know. And this first picture is taken in the early 1980s, and it's from Cold Spring Harbor. And at that time, well, we were young and beautiful, and things were different. But I've just started to develop this unique friendship that I had the privilege to have with Richard. So I was introduced to Richard via Mike Olston because I'm a virologist, and we were have a lot common interest in measles virus, and therefore I worked for a while in Mike Olston's laboratory, and then I was introduced to Richard, and for some reason we enjoyed each other company. We could have these very long conversations, and I could give it many examples on that. We met in different places, and my second picture is actually up in Lake Louise in the Canadian Rockies, and at that time we started to learn to know each other, and we discussed many different aspects of life. Of course, departing from the science, but we also went to an exhibition of various photographs, and where I bought this photograph that I have out in the summer house, and it always is a reminder of that. It's one of the first encounters with Richard. So he was really up to date in all the techniques that we're developing. We had been heavily involved in the use of monoclonal antibodies in order to dissect different viruses and identify their structural components. And the next step to that was to use synthetic peptides. And Richard, of course, quickly assured that that technique was developed at Scripps, and we could profit from that and learn how to prepare peptide and use it in our studies. One particular application that we have, when we talk about 1987, this was then when really the HIV epidemic was up and running, and I started to learn to know that there were in fact two types of HIV, HIV-1 and HIV-2. And HIV-2 was closely related to simian immunodeficiency virus. And it was also discovered by other scientists that there was an immunodominant site in the glycoprotein that one could be used for diagnostic by use of synthetic peptides. And in 1987, I was responsible for a conference up in San Francisco with all the luminaries of the HIV field at the time, with Bob Gallo, Luc Montagnier, Jay Levy, and others. And I knew at that time that Bob Gallo's laboratory had sequenced the SIV genome, the simian immunodeficiency genome. So I got access to that sequence and brought it down to Scripps, and then together with Richard and with Elliot Parks and some other people, we discussed if you could find the site, and we found the site, which was homologous to HIV type 1. And it is interesting because it actually, and all the difficulties to use this pointer, but in the sequence for the STLV3, as it's called here, you can see in the middle AFRQV. And this is quite illustrious because the virus comes from West Africa, and it's related to the SIV part of it. And now we use these two peptides, and since antipeptide, I brought them with me back to Stockholm, and then together with Gunnar Bieberfeld, who is on this paper, who had specific sera already characterized towards HIV-1 and HIV-2. We could do the serology, and rarely have we had such a very clear and distinct result. So one of the peptides did react with the HIV-2, and one reacted with HIV-1. And just one example of the use of synthetic peptides. So I continued to analyze this and developed a pattern during the early 80s of spending the month of November out in La Jolla. It is a month that you, in a way, can dispense with back home. It's dark and slushy, and whereas in some way La Jolla is always smiling, and you can start your day walking the beach and then do your work. So we did further dissection of this site by various setup of peptides, and I like to highlight this also because it's probably the last paper where I did most of the manual work myself, just to develop the dexterity. I always enjoyed to work with my hands, but later on, of course, other aspects came to interfere with that. But it reminds of a story. I remember when Mike Oldstone, when he didn't want to take the telephone, the saying was that he's pipetting. So we continue these very long discussions, and it's not surprising that I show you this, the Black Beach, and because at this time, in 1987, Richard told me on the way back from Europe in an airplane that he was going to take over after Frank Dixon. There had been a long regime of Frank Dixon, and now it was time for Richard. And the astounding thing, I think, is that he carried on this responsibility for 25 years. I mean, I know in many other contexts, it usually is enough some 8 to 10 years or so. If you have looked at the vice chancellor position or being the dean of a faculty or something like that. But Richard managed this in 25 years. And what also surprised me was when I came up to his office to visit him, it was usually rather quiet and calm. And later on, Paula Foley took over as a secretary. And she's actually in an early stage when the Department of Molecular Biology building had been built, and Richard had this idea that we should gather in a faculty room for drinks afterwards. So Paula Foley was also a self-taught bartender from that time. But it was something very quiet and calm in that office. And I've seen also all these many offices where the walls are full of all the diplomas from all the honorary doctorates or from prizes and so forth. Not in Richard's office. There were some selected pieces of art that he liked. Then you knocked on his door if it happened to be closed, or you just walked in and wondered how things. Oh, things are well. I mean, and there was always a unique atmosphere in there. And it's always struck me. I've seen so many of the people having this overriding administrative responsibilities, feeling harassed and running from one meeting to the other. Not Richard. So I asked him, what's the secret? I mean, how can you manage this? I mean, with all this enormous responsibility you have, and in such a calm way. Well, say Richard, I have this principle. When I start a day, I decided that's one particular problem that I need to solve. And once when I've solved that problem, then I relax. And it's a rare talent for leadership. And what about this room? It was rather unpretentious, little conservative furniture, and a blackboard. He used a blackboard. That's where you should stand. And Richard could ask, remember then, that I was not a part of the organization. He could discuss anything with me because I didn't have any kind of vested interest in this. So he could ask me, well, how does the PCR reaction work? I mean, could we review that? So we tried to understand it and did that. And then I said, well, let's take the walk. And this happened 2-3 times a week, actually. And this is a long walk. I mean, most of you must have done it several times. To get all around the golf course and then climbing up again. But it's time to talk. It's time to really learn to know each other. And I treasured those moments. But I'm still amazed that someone with that responsibility could do it in such a relaxed way. We did it in style also, and all of the emphasized staying at Claridge Hotel in London, taking the Concorde back to that. So there was always a feeling of quality in what we were doing. And the Palm Springs meeting, the desert meeting, what emphasized. So here was the joint between Richard Lerner and Bengt Samuelsson, who was the vice chancellor at the Karolinska Institute, and Paul Marx, who was heading the Sloan Kettering Institute. And these, it was a privilege to be a part of these meetings because they were always of high quality, but they were also very relaxed because these were people who enjoyed playing golf. And and if you're not a golfer, which I'm not, you can do your jogging or whatever. But also, there were amazing things. Richard brought me once to one of his golf club here, and he introduced me to the most famous Swedish female golfer, namely Annika Sörenstam. There was always the elitist, they wanted to the top when Richard was into this. But we did other things. So a small group of us was visiting Walter and Leonor Annenberg at the Sunderland estate. And never, ever in my life have I seen a private home that would come close to this. Here was all the original impressionists with Van Gogh and what have you. And all the American presidents have come there to visit, and a lot of royalties and so forth. A unique quality. And I remember my wife had some nice contact with Walter Annenberg, and we were exchanging letters later on. She was highlighting the Vikings from Scandinavia. So think about it. With Richard, there was always this quality dimension. In 1997, I actually left my professorship and chairmanship of virology at the Karolinska Institute to become permanent secretary, as it's called, at the Royal Swedish Academy of Sciences, having then the overriding responsibility for the Nobel Prizes in physics and in chemistry. I, of course, could not have an affiliation with Scripps, so I moved across the street into the Neuroscience Institute, as it was in those days. And this was then a visiting scholar with Jerry Edelman, another of those very bright scientists that came to locate to La Jolla. In fact, the powerful development of Scripps really was like a magnet because it brought fantastic scientists to this area. And I'm thinking about Manfred Eigen, and of course, and Escher Mose and Sidney Brenner and many others. You know, we had, when this building was finished, we had an inauguration, or Jerry Edelman arranged an inauguration. He was a very qualified violinist and apparently a personal friend with the Guarneri Quartet. And they were playing for us in this room. And you can imagine that this another high-quality performance as it was. Richard pointed out to me, well, it's I who have raised the money for these buildings. So it was not Jerry Edelman's achievement. Again, it was Richard. And how he could do that is just astounding. So I spent most of my time in the last 12 years writing books about Nobel Prizes and to understand how knowledge is growing and how it's expanding. And then the latest one so far is from in 2018, And as you see, that has to do with the genetic code. And in working on these books, I always have exchange with Richard and wanted his views on things. And we were discussing those that contributed to the genetic code prize. And one of them was Robert Holley, who got a Nobel Prize, in 1/3 of the Nobel Prize in 1968. And then Richard said, well, we have a statue of him, because after he had really made a contribution with transfer RNA, he was at Salk, but he was mostly interested in other things. So the ballerina on the right is taken on the kitchen table in the Lerner's home and ended up in this book, and the other one's taken in the library of the Salk Institute. So there is another book coming now in August this year, and you see, because the prices refers to genes, viruses, and cellular signaling. And the reason for this front page is, we discussed at the University of Washington in St. Louis. It's the couple, Gertie and Carl Corey, and she's the first woman to receive a prize in physiology of medicine in 1947. Now, in this book, I have a kind of a center chapter that I've called Advance of Knowledge is a Many Splendid Thing. And I talk about phenomena that have come back in the books over again. That is instructive and selective immunity. And I know that Richard would have loved this picture. He used it himself. He knew his history. And in that history, you can see Paul Ehrlich, the man with a dog in his lap. And it's also Svante Arrhenius, who's the first Swedish Nobel laureate from 1903. But he was a physical chemist. But he had a lot of ideas and was very argumentative. And I think it's an amazing picture, the way the two combatants, intellectual combatants here in this particular thing. And note this little formulation in the bottom here, corpora non acuit nisi fixate. Agents cannot act unless they are bound. It would seem like something very trivial. But what we're doing in whatever context, we talk about molecules that interact or don't interact. So it's a major truth in this, and we need to learn to know more about the specificity, the efficacy of interaction, and so forth. So, and of course, as has been already pointed out, one of the prizes that Richard got was the Paul Ehrlich und Dulwich Darmstadt prize. Then I've also written in this book about how you can come close, because you can imagine whatever Nobel Prize you get, there are several competitive groups, and eventually you have to make a choice. And by the way it has developed is that the practice is that you cannot have more than 3 recipients of the prize. In 2018, so half of the prize went to George Smith and Gregory Winter, and I'm not talking about what they do, but one can read in their in Winter's Nobel lecture, really that there were indeed parallel work of same caliber and so in the meantime, the group of Richard Lerner at Scripps and the biotechnology campus Stradian had developed an interest in antibody libraries. They took the next step and created libraries of heavy light chains pairs from the V genes from spleens or lymphocytes immunized with hapten. The pairings were generated randomly in the libraries referred to as the random combinatorial libraries. This is a pioneering contribution, and it's cited here. And you can see here also one of the critical references that were in. So I've written about Richard and written about that the unique quality of his work that was of a caliber that matches those that we have bestowed the prize. But at that particular time, we gave another prize, and that was this autumn, and I was writing this, and to Benjamin List and then to David Macmillan. And then I took notice, what was the key reference to Benjamin List's achievement? The key reference is the one where it's from the year 2000. Here's the first name, Richard number 2, and Carlos Barbas, already mentioned here, who sadly died at the age of 49 in this terrible thyroid cancer. So again, Richard had a fantastic feeling for knowing where science were going, and he was a part of that in so many different fields that I think it's hard to find other examples of that major achievement. So I had this email exchange. I wanted Richard to see the text that I was writing, and this is the last email that I received that Nikki could send to me from Richard also. Thank you for your kind thoughts. Hope you're doing well. All the best from us both. And that's how it all, to me, to have the privilege of developing a friendship with this remarkable man is something that I treasure and carry with me, and I express it with thanks to a good friend, an outstanding scientist, and an exemplary academic leader. Thank you so much.
Thank you. Thank you, Erling. So before we take a break, we have a little short presentation. So some of you might know that Gerli is the CEO of WuXi Pharmaceuticals, and he's also a member of our board of directors. So one of his colleagues is here, Lei-Tian Kuai, and he's traveled here to make a presentation to Nikki Lerner. So please.
All right. Hello, everyone. My name is Lei-Tian Kuai. I'm in charge of the DNA encoded library platform at WuXi, and actually thank Professor Baron and Derek for introducing the DNA encoded library technology and mentioning Dell Open. So today I wanted to pay a tribute to Dr. Lerner because this, I believe, this is a project that we are still working on. I personally, strongly believe in it. So it was in 2018 when we were building the platform, and we read about the comment that Dr. Lerner put in his 2017 publication, a review paper, and then he was suggesting that it is important for the industry to share its compound with the research institute, provide that to academic users, and then he suggested that Dell could be one of those technologies that could be shared between industry and academia. We are very inspired by this thought, and we decided that we want to take a crack at it. As most of you know, that this is not an easy thing to do between industry and academia. You will encounter a lot of tech transfer offices and lawyers in determining IP and money. And maybe it's a little easier for Scripps, but if you are a professor in Midwest and with 2 grad students, you're probably out of luck in terms of accessing industry compounds. So I emailed Dr. Lerner. I said, we have an idea. Maybe it can work because Dell is such a technology that basically anybody can run it on their bench. It doesn't require a heavy instrument, and so that maybe we could, set up a double-blinded experiment so that the researchers will receive the compound, run the experiment first, before we talk about contracting or money. So that was the sort of a core concept of Dell Open. And Dr. Lerner was very excited. I spoke with him. At that time, he just had a stroke. He talked very slow, but I can feel the strong enthusiasm of having this to happen. So he quickly invited many colleagues to form the Dell Open Scientific Advisory Board. And we had a couple of meetings. I still remember Professor DeWeck is asking all the sharp questions, and especially the, the first meeting we were left embarrassed because nobody knows how to do it right. And then it was unprecedented, either from a concept to the workflow, and how do we provide this to the academia. Even in the second meeting, we were still kind of tweaking, how does it work? But today, I'm proud to report, and so far, and we have been, doing very well. We have initiated 600 collaboration in the past 2 years, almost one collaboration a day. And we have 50 projects already at the finishing line with very good validation result. And then we also have several manuscripts, I believe, in review and should be accepted very soon. And the users, the academic users, no matter they are a big lab or small lab, from different countries, they can all freely receive this Dell Open kit that contains several billion compounds or DNA tagged. So with that, we have a small gift in memory of Professor Richard Lerner, and as you can see, this is on DNA molecule bound to a protein. And I hope this project will continue, and when we reach a critical mass, we may impact the field by an annotation of many targets and with many small molecules. That is the ultimate goal of the Dell Open. Thank you all.
So it is my distinct pleasure to welcome another brilliant chemist to speak on Richard's behalf. Ben was actually one of the first graduate students to ever come to the nascent Scripps graduate program. And safe to say that he never managed to reach escape velocity from this remarkable place. But his program involved trying to understand certain classes of serine hydrolases, and he developed that into a platform for chemical biology that has really revolutionized how we identify compounds as potential therapeutics or just even as tool compounds to interrogate biochemical pathways. He currently holds the Galula Chair. Bernie Galula was the founding dean of the graduate program at Scripps. He has proven to be a serial entrepreneur and has founded several companies, including the recently acquired Vividian Therapeutics. He was part of the team that helped initiate and fund the Abide Vividian Chair in chemistry and chemical biology. So please join me in welcoming Ben.
I have many vibrant memories of Richard, but none stronger than our first meeting. It was February 1992, and I had been interviewing for nearly 2 months at various graduate programs across the country. As someone with a visceral but still underdeveloped interest in science at the interface of chemistry and biology, I visited Scripps with much anticipation. At that time, Scripps represented the only graduate program in the world with an expressed commitment to chemical biology as a discipline. The founding dean of the graduate program, Bernie Galula, who is inseparable from Richard in my mind, escorted me to Richard's office and asked if he could attend the meeting. In retrospect, I think this was mostly for Bernie's personal entertainment, so I could see now how he anticipated what was about to ensue. Typical of Richard, there were no pleasantries. In his characteristic directness, he explained to me that the Scripps graduate program intended to create a new type of interdisciplinary scientist, one that would function like a stem cell, as de-differentiated as possible, such that they could tackle whatever important biomedical problems interested them with whatever methods were best suited to solve those problems. Only later did I appreciate that Richard intentionally used the term de-differentiated, not undifferentiated, as he was often quick to point out in our subsequent conversations that one of the biggest challenges he faced bringing scientists at any stage of development to Scripps was that they first needed to be, quote, de-learned, or stripped of their monolithic thinking and musty traditions that they had unwittingly carried from spending too much time at famous universities. before they could truly blossom in this special environment. Richard's words at that time hit home with me because I had just experienced exactly what he prophesied in my visits to other schools, where I was presented with balkanized graduate programs attempting to pigeonhole young scientists into committing their futures to the tiniest of subdisciplines. But before I could engage further, Richard switched topics, and that's when the real fun began. He next asked, "Where else are you considering going to graduate school?" And I responded that I was looking at Harvard. Leaning forward in his chair, like a panther about to pounce on its prey, Richard inquired, "To work with whom?" I mentioned a well-renowned immunologist, to which Richard abruptly exclaimed, "Why would you want to work with him? He's a bleeping MD." Now, having done my homework on Scripps, and Richard specifically, prior to our visit, I was both impressed by the forthrightness of his comments and also, admittedly, a bit bewildered. As Bernie and I left Richard's office, I sheepishly asked Dr. Glula, "Forgive me, but isn't Dr. Lerner also an MD?" To which Bernie grinned slyly, wagged his index finger, and responded, "Not that kind of MD." This story captures so much of what made Richard special. The vision to disrupt the status quo, the courage to communicate that vision with clarity and conviction, even when it required reinventing himself through shedding titles and accolades that had become, in his mind, irrelevant or even counterproductive to the mission at hand. The selfless generosity to invest his time in creating an environment that benefited so many others, and of course, the sophisticated and merciless wit. Yeah, that sense of humor. Richard was an incredible scientist and leader, and an even better human being. But what I remember most about him was how much he made me laugh, even if it was quite often at my own expense. I want to close by expressing my thoughts of gratitude. Reflecting back on those who have influenced my life, there are my parents and there's Richard. I owe Richard my entire career, from the moment I stepped onto the Scripps campus and into his lab, along with Jonathan Rosenblum. We were the first two graduate students in Richard's lab. Jonathan is here today, which makes me feel really good. He was there for us, giving just the right amount of nurturing, independence, cross-examination, and most importantly, trust. Trust that we could become impactful scientists. I often find that Richard is misunderstood by those who only superficially knew him. I'll hear them say, "Richard succeeded in building Scripps because he was an incredible fundraiser." But that's not what made Richard a great leader. I would even say that as visionary as Richard was, that was also not his unique gift. What was so different about Richard is that he combined an incredible self-confidence and competitive drive with a childlike curiosity to learn from others and a genuine interest in their success. In my experience, those traits are often mutually exclusive in an individual, especially if that individual happens to be a scientist. But not for Richard. I have never met someone so accomplished who took their greatest joy in the accomplishments of others. It is that feature of Richard's character that I try so hard to emulate each and every day of my life. So I'll now turn to my scientific presentation. I'm going to share with you one story today, and I wanted to share this story because I think it captures what Richard found so fascinating about science and chemistry in particular, which is when chemistry can illuminate a feature of biology that evades discovery through genetic approaches and more classical molecular biology approaches. We'll start with a very brief introduction to a technology that will feature throughout the talk called activity-based protein profiling. This technology really originated in our lab at Scripps in the late 90s, right after we started, and I'll give you just a few slides on its origins, try to mention a few people here in the audience or at Scripps in general that had a big influence over me at that time, and then we'll see that as Jamie, I think, mentioned, the origins really began with trying to expand what I would call druggable space within druggable space, which means trying to create small molecule probes and drugs for proteins that belong to families that we're pretty comfortable we know how to create small molecules and drugs against. But I'm going to spend the majority of the time on the second part, which is trying to extend those concepts to the broader collection of proteins in our cells that we may not have imagined years ago, we could target with small molecules. Something we refer to as mapping the ligandability or small molecule interactions of the human proteome. Let's see, we cut off a little bit, that's okay. The activity-based protein profiling technology is, at its core, quite a simple approach. We were in the late 90s, that was when the lab began. It was around when the human genome sequence first appeared. And we realized there were thousands and thousands of proteins encoded by our genome that lacked any functional assignment, any small molecule probes to study their biology. But we weren't without any information. We knew that they often clustered together into families that shared structural or sequence, or actually structural or functional homology. And so the concept was what maybe chemistry could exploit that feature. We could create chemical probes that could enter into native settings. This goes back, oh my goodness, this is a little odd. Okay, we'll do the best we can. This is not showing up quite right, but the chemical probes can enter into native settings and interact with hopefully a large number of proteins, and those proteins might share mechanistic or structural features in common. And we want these probes to find those proteins in any cell or tissue of origin. And then we can read out those interaction events. If the probes are fluorescent, by simply seeing them on a gel. If the probe is biotinylated, you can affinity enrich and identify the proteins by mass spectrometry approaches. And we hypothesized that if they could react with sites on proteins that reflect their function, they would provide a readout of the functional state of those proteins in cells. And we use a reporter tag to do that. And reporter tags include not only fluorophores and affinity handles, but also alkynes, which allow us to use Berry's remarkable azide-alkyne cycloaddition chemistry. And I will say that when this technology first began in our lab, we didn't really appreciate the broader implications. It was really individuals like Richard, Chi-Wei Wang, Paul Schimmel, and others that encouraged us to think broadly about this approach. And one of the areas that we began to immediately explore, which will feature throughout the talk today, is its use for ligand discovery, as Jamie mentioned. So in this format, you add a small molecule library to a cell. So unlike the DEL technology that you heard about earlier, we're going to use just cells and tissues as our sources of proteins, not purified proteins. And we're going to add a compound library to that system. And if one or more of those compounds happens to bind a protein in the cell, and that protein also happens to bind an activity probe, they'll compete with one another. And the small molecule could potentially displace the probe, and you can read that out in pretty much a variety of analytical formats. This has proven to be a rather remarkably versatile approach for ligand discovery, because it allows you to perform the discovery of small molecules against targets directly in native biological systems without requiring any sort of specific assay for your protein. In fact, the probe serves as a universal assay for essentially thousands of sites or thousands of proteins in the proteome in parallel. So each probe now, each site of interaction now can be assigned, even in cases where the protein has no other known biochemical assay format other than reacting with an activity probe. Over the years, our lab and many others have developed activity probes for well over a dozen, if not close to 24 major enzyme classes, and I'm not going to review this with you today. I just wanted to mention that we've been fortunate to be involved in a couple interactions throughout the years that have delivered clinical candidates for treating a variety of neurological disorders that those compounds were developed from their very origin using activity-based profiling as an approach. And these include inhibitors that target enzymes involved in regulating the endogenous cannabinoid system. So this is the system in your brain that produces lipids that act on the G protein coupled receptor that responds to tetrahydrocannabinol, the psychoactive ingredient of marijuana. And the hypothesis is if one can target these enzymes that regulate those endogenous cannabinoids, inhibit them selectively, raise those lipids up, they could produce a pharmacologically valuable subset of behavioral effects you see with direct cannabinoid agonists. Both these compounds operate as highly selective covalent inhibitors, including this very attenuated urea that inhibits the fatty acid amide hydrolase enzyme through irreversible chemistry, and this hexafluoroisopropanol carbamide, also another unusual electrophile that does so for the monoacylglycerol lipase enzyme. I want to pause here for a moment and mention that really the entire origin of these efforts, though, began with this work that I happened to perform with Richard and Bernie as a graduate student. And I mention this because I think at the time that this work was completed, it was pretty clear that it was going to set the stage for a lot of exciting science to follow. And all that work was done with Richard and Bernie, with their funding, and they had every right whatsoever to just take that work and go with it and tell me to go do a postdoc and find some other field to work in. And I think that's what like 99% of faculty would have done. They would have seen a new opportunity to launch into a field and go with it and tell the student, thank you for your efforts, go find someplace else to work in biology. But that was the opposite of what Richard and Bernie did. In fact, Richard pulled me in his office at this time because I was planning to go do a postdoc, and asked me the following question. He said, do you think there's something you can do in your postdoc that would be more exciting to follow up on your independent career than this work here? And I said, you know, Richard, not really. To be honest with you, I've always been fascinated by the interface of chemistry and neuroscience, and this is almost an ideal project. And he said, then take it and go. And it was just stunning to me, that level of generosity to not only support an independent lab, but to actually allow me to take the work that I had done in his group and launch our lab's independent career. So with that background, let's move on to trying to extend those concepts beyond the active sites of enzymes. I think if you asked Richard why Pfizer, our lab, and others had been so successful over the years creating highly selective covalent inhibitors for enzymes from a family like the serine hydrolases, he would say we cheat. And we cheat because the active site nucleophile in this enzyme family is so aberrantly reactive, maybe 7 orders of magnitude more reactive than a surface serine on your proteins. That gives the medicinal chemist a tremendous playground to come up with complementary electrophilic groups that can react with that hypernucleophilic serine and leave other residues unperturbed. When one thinks about extending the concepts of covalent chemistry beyond the active site of a hydrolase to other amino acids, you face a different opportunity and challenge. We're going to discuss covalent ligands that target cysteine today, and cysteine is the most intrinsically nucleophilic amino acid in our proteomes and proteins. But even the most nucleophilic cysteine is maybe only 1 order of magnitude more reactive than other cysteines across your proteins, which means you're not going to be able to rely on differential electrophilicity of groups to achieve specificity. The recognition component is going to have to be stronger. However, there was already some precedent that got us excited about this concept. For those of you who are interested in drug development, you'll know that virtually all of the covalent inhibitors that have been developed in the last decade or so targeting kinases to treat cancer operate by reacting with cysteines in the active sites of kinases. And what excited us about this work was that these cysteines are not catalytic residues. They're non-catalytic, or bystander cysteines, right? Which gave us the confidence that if one could find a pocket in a proteome that happened to have a cysteine residing next to it, that would give you an opportunity to create a selective covalent ligand against that site. And so this was really the origin of our work. This is how drugs like afatinib and ibrutinib work, by targeting non-catalytic cysteines in the active sites of kinases such as EGFR and BTK to treat cancers. So this led to the following platform that was originated with Aranthi Virapana, Chu Wang in our lab, and then pioneered further by Kerry Backus and Katja Vinogradova, who are all faculty around the world now. The idea is you're going to take small molecule electrophiles, add them into cells, and let them react with the cysteines they'll react with. These electrophiles look something like this. They'll bear electrophilic groups that are predisposed to react with the cysteine thiol, and variable recognition elements dubbed R1 and R2 here. Those will redirect, hopefully, those small molecules to different pockets in the proteome to bind to. And if those pockets happen to have a neighboring cysteine, these compounds would then react. And then you're going to chase those reactions with a very broad spectrum reactive compound like this iodoacetamide coupled to an alkyne here. This will react with thousands and thousands of cysteines indiscriminately in the proteome, except those that have been pre-engaged by these electrophilic fragments. They'll essentially punch holes in this gigantic profile of small molecule reactivity that can be read out quantitatively by mass spectrometry-based proteomics. So in this platform, you'll not only be able to determine the proteins that react with these small molecules, but the specific cysteine residues on those proteins that react. And Kerry really pioneered the first utilization of this, taking simple fragment electrophiles, adding them into our cells, and recording their interactions with cysteines on a global scale. And it was quite exciting, because even with a simple small library of a few dozen fragments, we identified hundreds and hundreds of sites of small molecule interaction in proteins throughout our cell. It was quite exciting. So for most of these proteins that harbored these druggable cysteines, this was the first evidence of their interactions with small molecules, as based on their presence or absence in databases like DrugBank. So you can see here, if you look at this pie chart, the smaller sliver of the pie, what you're seeing there is our efforts to rediscover proteins that are already known to interact with small molecules. You can see those are highly represented in protein classes like enzymes. These are families that we as chemists know with confidence we can drug. But the much larger slice of the pie, you're now seeing expanded representation of protein families like adapters, transcription factors, uncharacterized proteins, proteins that we might have viewed historically as being beyond the reach of small molecule chemistry. This is giving us some confidence that if one can combine recognition chemistry with covalent chemistry, you can breach the traditional boundaries of what we consider druggable. So this leads me to the story that I'd like to share today, which is an effort in collaboration with Janssen or Johnson & Johnson to discover small molecules that can regulate the androgen receptor in prostate cancer. So as many of you know, the androgen receptor is a major driver of prostate cancer. And this has led to the discovery of therapeutics that operate to treat this disease by antagonizing this receptor. One of them is shown here, enzalutamide, which is developed by Michael Jung's lab at UCLA and with Charles Sawyer, and now is a multi-billion-dollar drug to treat this disease. But like almost any cancer, prostate cancers will eventually develop resistance mechanisms against these drugs, and they're myriad and complex. They include overexpressing the receptor to high levels to override the drug action, creating splice variants of the receptor that no longer have the drug binding pocket, or in the most subversive mechanisms, creating gain-of-function active site mutations that convert these ligands from antagonists of the receptor into agonists. So this has led Janssen and other companies to try to come up with new ways to regulate the androgen receptor in prostate cancer. And they developed this very interesting high-throughput screening approach to measure the messenger RNAs for the androgen receptor and its major splice variants in a way that could be screened for small molecule effects. And so we took our electrophilic cysteine-directed library and performed that screen with Janssen. And we were fortunately able to find some interesting hit compounds, including this chloroacetamide B21 shown here, which substantially reduced the androgen receptor transcript in prostate cancer cells. And if you followed that effect over time, you'd also see reductions in the protein level of the androgen receptor. What piqued our interest was to follow the kinetics of this phenomenon. If you do that, you see that the messenger RNAs for the full-length form and the splice variants of the androgen receptor decrease first. Then the protein goes down. So that suggests to us that whatever the mechanism of action is here for this small molecule, it operates on the transcript regulation of the androgen receptor, and then indirectly, after the mRNA goes down, the protein is decreased. Here we were very fortunate that the structure-activity relationship for this effect was exquisite. Modest changes to the compound would either increase activity or completely eliminate activity. And what was especially gratifying was there was a single stereocenter in this compound, and that proved to be critical for the activity. The R enantiomer was active, the S enantiomer was inactive. So why this is important is this gives us a set of tool compounds to compare in their proteome-wide reactivity to deduce the mechanism of action, which should be a protein that reacts with all of these active compounds and none of the inactive controls. And we're going to use this cysteine reactivity platform that I mentioned earlier to achieve this objective. This platform can profile thousands and thousands of cysteines in our cells in parallel. And you can display those, say, in a heat map to look at the cysteines that are reacting with your small molecules. And most of this heat map is white because of these 20,000 or so cysteines, most of them are not reacting with these small molecules. Instead, we want to focus on that little blue area, which are the cysteines that are reacting with our small molecules. And we want to focus on the cysteines that are reacting with the active compounds, but not the inactive controls. And there really was only one cysteine that satisfied that requirement, this cysteine 145 in protein NONO. This is what happens when you let someone else put your slides on another computer. That's okay. I'll walk us through this. It'll be fine. What is NONO? So NONO is an RNA binding protein. It's a member of a 3-protein clan of sequence-related memberships that form homo and hetero oligomers. Despite sharing about 70% sequence identity, only NONO has this cysteine, not its sister proteins, PSPC1 and SFPQ. And that's extremely important because that's going to help explain how chemistry here differentiated genetics as we move on through the mechanism. Also interesting was the presence of this cysteine in the structure, located right in between the 2 major RNA binding domains of NONO, suggesting it might be at a site that was involved in regulating RNA binding. So NONO and its sister proteins have been implicated in pretty much every step of mRNA maturation and processing. So we thought this was interesting, right? We had discovered a small molecule that regulates the androgen receptor transcript in prostate cancer cells, And the major target that looks responsible for the mechanism is involved in binding mRNAs, right? So it all seemed to be coming together quite nicely. So Stefan Kaufman, the postdoc who really led this work in our lab, did what I think anyone else would do at this point. He used genetic methods to knock NONO out under the assumption that if we knocked it out genetically, it would replicate the effects of the compounds. And that's where we got our first interesting surprise. This is a Western blot showing the androgen receptor expression in NONO wild-type and knockout cells. And what you can see with the red arrows is that when you knock NONO out of these prostate cancer cells, the androgen receptor doesn't change at all. So this indicates that the genetic knockout of the protein does not replicate the effect of our small molecule. Instead, what it does is it completely blocks the effect of our small molecule. You now see that the ability of our compound to suppress the androgen receptor is lost in the NONO knockout cell, giving us the first clue that these compounds acted through a mechanism that required NONO, but was not replicated by the loss of NONO itself. And you see in these contexts without NONO present in the cell, the active enantiomer looks just like the inactive enantiomer. And you can see the exact same thing at the mRNA level, where the mRNAs for the androgen receptor and its major splice variants are suppressed by the active compound only in wild-type cells. When you knock NONO out, no such effect, and knocking NONO out alone has no effect on the androgen receptor transcripts. Understanding now that NONO was required for the mechanism but did not replicate the effects on its own allowed Stefan to do another interesting experiment, I think, to really nail down NONO as the relevant target. And that is to reintroduce wild-type NONO into the knockout cells. That restores the active compound's activity. Instead, when you reintroduce another variant of NONO, where the cysteine that our drug is acting is mutated to serine, making the drug inactive, that variant of NONO can no longer restore compound activity. I think really providing strong evidence that these compounds act through NONO, through cysteine 145 specifically, to suppress the androgen receptor. Okay, so let's pause here and take you through a few more slides of how we understand the mechanism. What we can say at this stage is through high-throughput screening with Janssen, we've been able to discover an RNA binding protein that had never been known to interact with small molecules before, that can be drugged at a specific cysteine to suppress the levels of this oncogenic transcription factor, the androgen receptor, in prostate cancer cells. We have some interesting questions we'd like to ask. For instance, what other types of genes and proteins can be regulated by these NONO ligands? And you can do that now by global profiling, such as RNA sequencing. And when you do that experiment, it's quite fascinating. This is cut off, unfortunately, at the bottom, but I'll walk you through it. There's a discrete set of transcripts, including the androgen receptor, that are all regulated by the active NONO ligand relative to its inactive enantiomeric control. That's what you're seeing on the x-axis. What you're seeing on the y-axis is another interesting comparison, which is all the transcripts in the cell that are regulated by the active ligand in wild-type cells, but not NONO knockout cells. So this one-to-one correlation that you see here indicates that virtually all of the effects of these active ligands on transcription occur through the NONO protein. And you can see that quite nicely with the androgen receptor itself. You see quite nicely the androgen receptor goes down. In the wild-type cells treated with the NONO ligand, that's blocked by knocking out NONO, which has no effect itself on androgen receptor. What about the molecular and cellular mechanism? Here we got clues from two separate studies. First, when you image where NONO is located in cells, what you see is these active ligands promote the clustering of NONO in the nucleus into these little speckles or foci or condensates. These are thought to be RNA protein interaction hubs, which gave us the thought that maybe what these ligands are doing to affect androgen receptor transcription is they're stabilizing NONO binding to its mRNA substrates. And indeed, working with Jean Yao's lab just down the street here at UCSD, who studies RNA protein interactions on a global scale, using some of his methods, we were able to show that the NONO ligands promote the binding of NONO to its mRNA substrates. So this is a case where now the ligands are binding NONO and trapping the protein, if you will, on its mRNAs. That's one mechanistic insight. The other comes from what I brought up at the very beginning, which was why is it that the genetic knockout of NONO does not replicate this phenomenon? And that comes from looking at its sister proteins, for instance, PSPC1. This is a protein, remember, that looks just like NONO, but lacks the cysteine that the drug acts on. And remarkably, when you knock NONO out of cells, the most dramatic change in the proteome is the upregulation of its sister protein, PSPC1. This has led others to speculate that this is a compensatory mechanism. The cell senses that NONO isn't there, its mRNA profiling is not being managed properly, so it upregulates its sister protein, which goes on and does the job. I think even more incredibly, within 24 hours of treating cells with this no-no ligand, the same thing happens. PSPC1 goes up. So the cell is somehow able to tell that no-no isn't functioning properly when these ligands that bind no-no are present. But the problem for the cell is no-no is still here in this instance, bound to our compounds, right? So even though it's upregulating its sister proteins in what I would consider to be a futile attempt to compensate, the inactivated no-no protein is still present. So this has led to a model referred to as trapping. So let me walk you through it. So in a wild-type cell, no-no functions in collaboration with its associated proteins to regulate mRNA processing and transcription. Everything is fine. On the far right for you over there, the no-no knockout cells, this PSPC1 sister protein gets overexpressed, replaces no-no, and now the cell is fine again. But when we treat with our ligands, PSPC1 goes up, trying to compensate, but no-no is still present, trapped on the RNA, and the compensation is blocked. And this is, for those who are interested in drug discovery, you'll know that this is somewhat analogous to how PARP inhibitors operate to treat prostate cancer and certain breast cancers where the BRCA1 mutation is present, trapping PARP on DNA to create a greater effect than that which is observed in a PARP1 knockout. Finally, as you might expect, if you treat these cells long enough with no-no ligands, you will actually see those cells undergo a blockade in their growth. And it's only the wild-type cells that show this blockade of growth. The no-no knockout cells are resistant, right? So that's consistent with this effect occurring through no-no. So this is the story I wanted to share with you today. I think the other really interesting nugget about this seems like a lot of work, right, and pretty exciting. And what we got back from Janssen was, way too early to develop this ourselves, right? So despite having discovered a mechanism that can suppress the androgen receptor, the exact target they wanted to go after in prostate cancer, they said, too early, keep working on it, right? And this is where I feel like having, as Phil brought up earlier, Richard as a guiding light to say that's exactly what you want, right? You don't want the pharma company to get involved at this stage. They'll just mess it up, right, through some conservative bureaucratic process. So in some ways, one could look at that outcome and say we're liberated to continue to study this process, take it further ourselves, maybe even into a biotechnology company that can be purposely focused, specifically focused on this objective. I mentioned Stefan and Garrett Lindsay, the other student in the lab who have driven this work. I wanted to finish here by just mentioning that I think the greatest tribute that I can make to Richard in my career is to do for others what he did for me, right? And so you will never hear me ever tell someone in our lab, you can't work on that when you leave our lab because that's our work, right? Anyone in our lab can take anything they want and work on it independently. I encourage them to do so because that is the way to pay forward what we received, Jonathan and I and others who have trained with Richard in our careers. And so I'm forever indebted to him, not only for how he trained me in the lab, but how he trained me to be a faculty member who gives more than they receive. So thank you for allowing me to speak here at this meeting. It meant a lot to me.
Please welcome Doron Shabbat. He is a professor at the School of Chemistry at Tel Aviv University, where he holds the Americo Latte Chair of Chemical Processes. His research program is chemical biology involving self-immolative molecular systems involved in drug delivery, developing long wavelength fluorescent probes for imaging, and creating chemiluminescent probes in aqueous media. And on the basis of his work, the Israel Chemical Society awarded him the ICS Prize for Outstanding Chemist in 2020. So in a minute, we'll hear from Doron, but please join me in welcoming him.
Okay, so my talk will be also scientific, but a bit more intimate. And first I want to thank Pete for allowing me to speak today. It means a lot to me. As I will show you in my presentation, Richard has done a thing for me that I consider as extraordinary or unbelievable. I want to tell you my story and how I actually arrived to Scripps and how I made the contact with Richard. So it's all start in August 91. In August 91, I'm a PhD student in the Technion under the supervision of Professor Udi Kenan, who is here in the audience, titled Catalytic Antibody. And Udi can tell you his story about how he arrived to Scripps and for a sabbatical year that later extended to almost a permanent job. And I'm a junior student, a PhD student, just a few months before Ben even arriving, and Phil Dawson was not, even not here yet. And at that time, Scripps looked different than it is today. Scripps was only these two buildings, the immunology and the molecular biology. Beckman is not existent at that time. And while I'm coming here with Udi, two other groups are joining Scripps. Barry Sharpless is coming to Scripps, and Dale Boger also is coming to Scripps, and you need to find space for them, and there is no space here, so we were all located in this building. This is Scripps, this is Corset Street. We are here now, and this is this building. This is the CVN building, the Blake building. I'm right? Okay. And yeah, so we are all located there, and that's how this is how the building look today, and that's where we started. And this is me in 1991. This is the CVN building, and we are all starting to work there shortly after even Ben arrived to work there, and he worked for a while. Phil Dawson was working with Steve Kent just across the hall of my lab, so we were all here a long time ago. Phil Barron was 12 years old at the time? Yes. Okay, so you are not here. This is my first Scripps ID badge from, look at the date, August 91. And I guess you can all recognize this young scientist. The reason that he's sitting on the floor, because this is a new building. There are no furniture yet. And Udi took this photo in 91. But just to explain you about the atmosphere at the time, when you see Barry Sharpless sitting on the floor and you're a young student, this gives you a huge push of motivation. If you can see the floor, then you can stand on your head and do the chemistry that they ask you to. And this was the atmosphere at that time. Here's Dale Boger at that time. So the picture is a bit blurry, and you can see Udi here. So this was a great time here in Scripps doing science. And between 1991 to 96, I actually shuttled in between Israel and here, and we are part of the Lerner group at that time. So my interaction with him is relatively mild. I'm a junior PhD student, and but of course I met him on several occasions. But then in February 97, there's an annual meeting of the Israel Chemical Society, and it's a joint meeting with a Scripps Research faculty. Some of this faculty are here today with us, and let's see them. So you can see Chiwei and Casey. Chiwei hasn't changed. He looks the same, I think, today. Here you can see Ian Wilson and Peter Wright. And here you can see me and Barry Sharpless, and here is Udi on the side. And in the last photo from this series, you can see me and Richard. So obviously, well, he knew me. We never talked intimately, but he knew me. And then something unusual is happening. And this is what I call the game life-changing event of my life. And to explain why I call it this, I need to expose some personal information about me. I have a very strong attention deficit disorder. Even now, when I'm talking with you, I'm fighting with a lot of images in my mind and try to stay focused and to tell you what I want to tell you. So I was able to do the science in the lab, but I couldn't really do writing or analyzing information in a way that usually a good PhD student is able to do, and I was very much insecure myself. So although I really wanted to continue and to be one day a group leader in the academia, I was so insecure of myself, and I already had I decided that I'm not going to continue, and I had a job in Israel in some biotech company. I signed the contract. I was about to move to a different city, but on that day, Richard is in Israel, and it's noontime. He's tired. He's in a jet lag, and Udi is asking me to drive him to the hotel for rest. So this is the hotel that we are going to, and he started talking to me. In the way, you realize that I have 2 Nature's paper in my PG. Let's face it, I was a student. These are Udi's paper, not mine, but I was the student that did the work. Actually, Pete reviewed one of these papers. I remember him telling me about this during the Wolf Award ceremony. And it was in the field of catalytic antibodies. So Richard, of course, when he was talking with me about that, you know how Richard is working. He immediately said, OK, stop the car. Let's go for a coffee inside the hotel. And they said, OK, what are you going to do now? You're going to do a postdoc, going for a company to work. Why don't you just come back to Scripps, stay a couple of years, and then go back to the company and see what if it fits to you. But give yourself a chance. And I told him, but I already signed a contract with a company, and I rented an apartment. So this is no problem for Richard, of course. So he's saying, OK, how much money did you spend so far? I told him, at least $2,000. Yeah, OK, so you got $3,000 immediately, you say, and just finish everything and come next week to Scripps. And this is how I find myself in April 1997 as a postdoc with Carlos and Richard here in Scripps, and I'm starting my academic dream. And Richard really believed in me and gave me whatever I wanted. And of course, at that time, we were working in the field of catalytic antibodies, and the classical approach was that the transition state analog stabilization is being used in order to elicit the pocket in the antibody molecule. And when I came to Scripps, they already have this technology that Carlos came with, the idea of the reactive immunization. This 1,3-diketone that is attached to this amino lysine, and you can actually get this antibody that was designed originally to do aldol addition reaction. 2 antibodies were generated in this pool, 38C2 and 33F12, and you can see here the X-ray of the antibody and the lysine that is buried deep inside of hydrophobic pocket. Here's another X-ray structure, this time with the diketone. This came much later, and an absolute proof that you can elicit this lysine catalytic residue inside the pocket. And I was immediately assigned to work on that, and this aldol reaction that was originally done before I arrived to the lab was just the beginning. Actually, you found that this antibody can catalyze a reaction with numerous substrates, you can change the donor and the acceptor and get all this molecule with very high enantiomer selectivity. And the success of the antibody made it as the first, or the only, I think, commercialized catalytic antibody that was sold by Aldrich at that time. And when Richard get success from his group, he immediately want to share it back. And here's an example for what is happening. September 18, 1997, dear Doron, here is a little gift on the occasion of the first commercialization of the catalytic antibody. This won't have been possible without your help. Warm regards, Carlos and Richard. And they gave us some extra cash money beyond our regular salary. So that's just one initial example for the generosity of Richard. Anyway, let me go back to the science. So the antibody could also catalyze the retro-aldol reaction. And here's an example of aldol that react with the lysine. And in this reaction, it releases this specific ketone. This specific ketone has the beta position, a leaving group. And then we found that the antibody can also catalyze the beta elimination or the retro-Michael reaction of this and release this small red molecule here. The importance of this is that you were able to expose a hydroxyl group. And if you expose a hydroxyl, it could be also an amine group. This means that you can use this sequence to protect molecules like alcohols or amine and release them via the sequential retro-aldol retro-Michael reaction. And with that, we turn to the idea of prodrug activation. Here, this is doxorubicin, a standard chemotherapeutic drug, and you see that we protect this amino sugar with this retro-aldol retro-Michael protecting group, and the antibody can catalyze the sequence of two reaction and release the free drug. And then the interesting and advantageous thing in Richard and Carlos' group that chemists could collaborate with biologists. And there is a biologist here in the audience that is an expert in antibody, Christoph Rader, and I was working with him on this. The idea was to combine two antibodies to form a bispecific antibody, one with a catalytic arm and one with a binding arm. And here is the illustration of the idea that we wanted to achieve. The bifunctional antibody will bind to a tumor cell, to an antigen on a tumor cell, and the catalytic arm will cleave or activate a prodrug in a catalytic manner and generate the cytotoxic effect of the drug. I want to show you an animation movie from 1999 that Richard really loved. And this was, at that time, video was not so developed as now. So in this video, that there is all, this is Christoph, and I'm supposedly injecting the antibody, and you can see the bifunctional antibody running inside the blood vessel to a tumor site and locked on the tumor. Look at the animation from 1999. And here's the lock. And then you can see the catalytic arm activated the prog via the sequential retro-aldol beta elimination sequence. And in this way, you activate a numerous molecule of drug. And at that time, when we were talking about the ADC antibody drug conjugate, people did not believe that it's even possible that such compound will arrive to the clinic. As here you can see the chemistry that I was just describing. Anyway, so this was the work with the antibodies. And another thing that we can achieve with this sequence is actually, again, by exposing here a functional group like hydroxy or amine, we can actually generate a reactive molecule that will emit light via fluorescence or other mechanism. And this molecule emit light via chemiluminescence. I will later talk about this molecule that I start working in my independent career about. But here we had a problem. This molecule is too big to enter the antibody active site and could not reach the catalytic site of the antibody. And then we start working on developing special linkers that will translate the cleavage event here into release of a molecule here. And actually, this affected a lot later my independent career. And this is what then we came with the concept of self-immolative linkers that were known, but we translated it to systems that were not generated so far. And in this case, the linker simply is a special spacer that once you cleave it, the cleavage site here is translated to a cleavage site here, and the free drug is released. So at February 2000, sorry, I'm getting a position in Tel Aviv University, again, not without the help of Richard. Richard traveled to Israel, went to this university to give a presentation, met with the head of the department, and made sure that they are giving me the position. Simply as that. There was no, for him, there was no question. But as I told you, I was so insecure of myself, and he always comes down. He said, look, you go there, but you keep returning back to Scripps whenever you want. And I started my independent position on the field of self-immolative linkers that I just explained you. And the first idea that I was thinking in the concept that was shown before in a prodrug design here, as you can see that this is a cleavage site, and when you release, you have this 1/6 elimination of to generate an azaquinone metide species and a release of a target molecule. In this case, it's just a model for nitroaniline. So the first thing, idea that I have in mind is to combine this 1/6 elimination with 1/4 elimination, because this elimination can happen to both sides. And with this combination of these two, you will generate a dendron-like molecule, an AB2 molecule that once you cleave this substrate, the two drugs will release from the same molecule. And it will work like this: cleavage of the substrate, obtaining of the free aniline, 1/6 elimination here, and then addition of water, and elimination to the other side. So basically, you reach here in an amplification effect, and you get a branch molecule. And if you have a branch molecule, you can build trees. And then we came with the concept of self-immolative dendrimers. The idea is that you cleave once, and you have a domino-like reaction that collapses your platform to release all the end molecule from your system. And here's animation of what we have achieved at that time. And only recently, in collaboration with Merck Darmstadt, we now have a system of an ADC, an antibody drug molecule, that, as you can see here, it has a binding site through malamide chemistry to cysteine. And this is a general cathepsin B activated product system. But the idea that you can either double the amount of drug that your system carry, or you can use two different drugs that act by synergism and different mechanism. I continue to work on this idea of self-immolative molecular system, and we later developed the system that we call a self-immolative polymers. I'm not a polymer chemist. We just developed it and left it. And a lot of groups from over the world that do polymer chemistry enter to the field and came with either a new example or use our own chemistry. But I want to go back now to this light emitted molecule that I just described. This is that I worked with Richard and Carlos in 1999. here you can see the mechanism of the light emission activation. You remove a protecting group, you generate a phenolate, and now nobody really understands, but there is some electron transfer probably to the sigma star orbital of this peroxide that cleave this molecule into two parts, and then you get this benzoate in its excited state. The benzoate decay to the ground state by emission of a blue light, and if you take, for example, silyl protecting group with fluoride, you get a nice emission. At that time, we used the retro-aldol-retro-Michael linker in order to see if we can generate an assay for an aldolase antibody. The problem with this assay that the light emission properties of this molecule are quenched by water in water. It's like 10,000-fold than the original light emission if you do it in a solvent like DMSO. So when I wanted to demonstrate this to Richard, the potential of this molecule, I did it in DMSO, and with your permission, I want to show you a demonstration of this here on the stage, and Summer will help me, because this was a really phenomena that influenced a lot my scientific life later. So what he's going to do is going to take this molecule with a silyl protected group, and instead of using KOG-DMSO, he's going to put a TBUF, but you can see that there is light even with this illumination. The light that is emitted from this molecule is really tremendous, but this is in DMSO. If you would, I don't want to kill the light, but if you would add like 1 drop of water inside, all the light will be diminished. You can leave it on the stage until it is over. Thank you. Summer is currently a postdoc with Phil Barney, and he graduated in Tel Aviv University in my group. And he has a position now in the Weizmann Institute. He will be leaving soon there. Anyway, when I show this demonstration to Richard, and we got this blue light that you can see here, but we use KOH, of course, not an antibody, and we did it in DMSO. So I'm showing this to both Richard and Carlos, and Richard's response was like this when he looked at Carlos, he said, who needs an antibody when you have KOH? That's an example for Richard's sense of humor. But this was done in 1999, and only 5 years ago, 20 years later, I understood where is the problem. The problem here is simply that this excited benzoate is quenched by water through some kind of energy transfer, either through rotational or vibrational levels of energy in the molecule. And we were trying to solve this problem, and the idea was that in case of generating this benzoate, we will generate this benzoate, the one that has electron withdrawing group with a donor on this molecule. And anyone that understands something about fluorescence will tell you that when you have a donor and acceptor functions on the same molecule, there is a good chance that it will be an emissive molecule. And that's the thing that we're trying to test, and if this would work, then dioxetan like this, when they decompose, will release this emissive molecule, and this will emit efficiently in water, and we can get the reaction that we saw there in water. So we synthesize 4 dioxetan molecule, and here you see a methyl acrylate acceptor. Here you have methyl acrylate with addition of a chlorine. Chlorine also withdraw electron and decrease the pKa of the phenol. So if you do it in physiological pH, you generate more phenolate. And here you have acrylonitrile, a stronger acceptor, and the one with chlorine here also. So taking these molecules, we compare it. The light emission, this was non-compound, commercially available, you can purchase even, and you can see in water, this is the light emission that is measured very low, and only by adding methyl acrylate, you can have a 700-fold increase in light emission in water, and by the chlorine, with the chlorine is 718, going to the acrylonitrile, you're over 2,000, and again with the chlorine, 3,000 fold effect. I think this thing happened once or twice a life to a scientist in the lab, that you're trying to improve something and you prove it by a 3,000-fold. So for the first time, we have now a molecule that can emit light efficiently under aqueous condition. And we use a beta-galactosidase substrate to demonstrate. This was, again, commercially available. This is our molecule. And you can see that the light emission that is coming from our molecule is significantly higher. The inset here is describing this flat light that is coming from this molecule. This was known. So we really were able to get, for the first time, a molecule that can use as a sole component in a biological system. And that's important because when they use this molecule, they always add additives that are not compatible with cell. They were toxic to cell. And with our molecule, we can use it as a sole component. And for example, for cell imaging, chemiluminescent cell imaging was never demonstrated. These are bioluminescence that was done with luciferase. But we could take our molecule and with this extraordinary high quantum yield and image cells. And here we take a cell that express a certain, that have the Laxi vector that express the beta-gal enzyme. And you can see that the light, the cell were illuminated. When you take the wild type, there is no light on it. So this was illumination of a cell that was transfected with a vector, but it doesn't have to be transfected. We can do it for any enzyme that exists in the cell. So this is a system for cathepsin B, for example. So here, this is only for solubility, and here you have a cleavage site for cathepsin B. And then when we took a cancer cell line like leukemia, you can see that we can image it with this cathepsin B probe. I will skip this example because I don't want to go too deeply in the science. By the way, the technology was commercialized by a Swiss company. And again, everything that I do is connect with Scripps, because the founder of this company is a former postdoc of Julius Riebeck. And he's in my age, and he was not a Scripps. It was Julius before Julius arrived to Scripps. I wanted to end with just another example that demonstrated collaboration with scientists here in Scripps. And this is regarding the synthesis of this molecule. The synthesis of this molecule was developed by scientists much before we entered to this field. And here's a classic synthesis. Pay attention to the number step, 1, 2, 3, 4 step of synthesis. Not very friendly synthesis, especially this step that you use titanium chlor-3, highly explosive, or not explosive, but inflammable, I would say, the one that catch on fire. And that's not a very pleasant synthesis. And then someone developed another synthesis that has several steps. And when I talked about this with Phil immediately understood that we're approaching the idea in a wrong manner. And most of you know Scripps, and you will agree with me that you know one thing or two things about synthesis. And Phil immediately redesigned the synthesis and came with this idea to use a CC bond coupling sp2 in order to generate fast and rapid libraries of molecule. And this will be based on a late-stage functionalization, protecting group free, and a promiscuous scope, exactly like what Phil liked to do. And we developed this chemistry. We prepared this Steely reagent. And with this Steely reagent, we can do cross-coupling to various haloaryl bromide, and then oxidize them and get our molecule much easier. Here's just one example. This molecule was a very good emitter and 90% fluorescent quantum yield that also affect the chemiluminescence, and we can get long long life, also long life emission profile from this molecule. And when we synthesized this molecule in traditional well, it was 10-step synthesis. And with the new synthesis that we developed with Phil, 1, 2, 3, just an example. But of course, this is just one example, and we prepare a library of molecules like Phil is always exemplify in his work. And this work was just recently published in Angewandte, and we discovered numerous of new dioxetane. In the last 5 minutes, I want just to go back again and to expose personal communication with Richard that I have. And again, I need to go back to my limitation. The reason I always needed the support, I was so insecure of myself that in every step that I did, I needed the support of Richard, and he completely supported me in everything that I did. And so I had to share with him every step that is happening in my career. For example, the first, okay, this is how it was working. I was usually writing an email to Paula that I would like to visit in Scripps. And in this case, I also, as you can see here, really wanted to show some gratitude and ask him if I can invite them to a personal dinner. And she would reply, yes, you're confirmed, you can come. And we can also arrange dinner with the Lerners. And Nikki, I don't know if you remember this. This was back in 2004. And here's the dinner that we had together in Carlos' house. And I really feel lucky that I could have the chance to do this. And as I told you, I share with Richard everything that happened in my career. For example, this is the first paper that the first significant paper that I published in my independent career. Here, look at the text. This is from 2002. I recently got a fax from Dr. Peter Gerlitz informing me that my paper was accepted. And I had to thank him personally about this. This is maybe the last personal note that I wrote to him. In 2009, I became a full professor, and I also had, of course, to let him know and to know how much I owe him for what he was doing to me. So here's just a story of one episode. Phil presented this restaurant that was his favorite. This was always an adventure to go to dinner with Richard. You're sitting with him on the table, and you look in the menu, and he's ordering for you. And you usually choose interesting food, I would say. And then I see him also ordering food that is not in the menu. And the waiter was simply preparing whatever he was asking. And in February 2000, when I finished my postdoc period with him, and I was about to go to Tel Aviv, I remember this dinner very clearly. I was kind of, it was dinner with him and Carlos, and I was kind of trying to understand how did he gamble on me and find me suitable to come to work for him when he didn't know anything almost about me. Richard was looking at me and saying, look, you're making a big deal out of nothing. You drove me to the hotel. I saw the way that you are driving. If you turn to be a bad scientist, I could always use you as my driver. Yeah, this was Richard. I think I want to end with this slide maybe. I believe that I honestly earned the title the most beholden person to Richard. I clearly owe Richard my scientific life. I would not be a university professor without him. There's no doubt in my heart about that. And here is just a last slide that I call it Science and Joy with Richard. You see Richard playing golf and one of his cars. We talk a lot about cars. And this is a visit of Richard in Israel in one of the Arab villages in the north of Israel. Udi was generous enough to give me this photo when Udi took him to a tour. And maybe the last story, because I think I have another 2 minutes that I want to end before I finish my presentation, is another typical crazy story about Richard. I think the year of 1999, the former Prime Minister of Israel and the future President of Israel, Shimon Peres, is arriving through Udi to Scripps to talk with Richard about building an academic scientific institute somewhere in the Middle East. Udi can fill you up with the details if you want. So this is really a meeting that dictates the future of the Middle East and maybe the rest of the world, if you think that the Middle East have an impact on the rest of the world. And they're sitting in Richard's office and talking. Richard gets bored after a while. He's stepping out, and he sees me working in the lab. He's catching me with my arm and say, come, I need to do something with you. And he's going back to his office. The security don't let me enter. Say, why? But he's just pushing security away. This is the former Prime Minister of Israel. He's pushing them away. And we are entering to his office. Udi is there talking with him about building this institute. And Richard stopping the discussion and say, Shimon, look, this is Doron. He's a postdoc from Israel working for me. I think you should talk with him. I see Udi is turning white. He will not admit with that, but he was turning white. And it happened that I was sitting there for a minute, and I understood myself that this is not my place, and I left, of course. But again, just an example the way that Richard is pushing things and working in the way he's working. I will, of course, be always indebted to him, and I will owe him and remember him in a way that nobody else affected my life like he did. So thank you for you guys listening to my talk, and here I finish.
Okay, so thank you, Doran. And we should start the computer swap back while I introduce Professor Wilson. So Ian is the chairman of the Department of Integrative Structural and Computational Biology here at Scripps, where he holds the Hansen Chair. So I'm a member of his department, and so I have an unusual relationship with Ian in that I'm his boss and he's my boss. And which hat he's wearing depends on whether he wants something from me or I want something from him. So Ian has made multiple seminal contributions to the structural biology of the immune system. He's determined structures of antibodies, the T cell receptor MHC complex, and most recently, he's been focusing on the recognition of viral co-proteins by the immune system. He's made huge contributions to the study of viruses, both HIV, influenza, and again, most recently, the coronaviruses. He's got a remarkable career of making highly impactful discoveries that really affect human health. And he's been a terrific department chair and has been instrumental in recruiting all kinds of young faculty to the department. So he's obviously worked closely with Richard for much of his career, and we look forward to his presentation. Please join me in welcoming Ian.
Okay, I'm going to start my story with Richard when I first met him. And that was actually in Salt Lake City. Oh, I've got my mask on. So. And I shouldn't have actually been at this meeting. I was at, the person I was working with at the time in the lab at Harvard was Don Wiley, and he was supposed to go to the meeting, and he couldn't attend. And so he said, well, why don't you just go to the meeting? So I set off for Salt Lake City, and I stayed in this hotel here, the Park Hotel. And as you can see, it overlooks the temple. And my bedroom window overlooked the Archangel Moroni there. And I thought to myself, this is a sign. Something's going to happen. And I didn't know what it was, and it was going to be Richard. And so I went to this meeting on genetic variation among influenza viruses. And the reason I was there was because I had been working on the structure of influenza virus hemagglutinin. And this is the major surface antigen of influenza virus. It's in green up here. I don't think this pointer works very well. So this is it here. And we had been working on the structure of this. And as you can see, we didn't have very great graphics in those days. These were all hand-drawing pictures. And this one I drew by myself on the left, and I got somebody to help with that on the right. And so what we had discovered in that was, what did the structure of this green blob look like? And how did the immune system respond to influenza virus? And by that, we had some sequence information. And that sequence information was on natural variation from when this virus came into humans in 1968 up to 1975, and also some lab variants. And so on this schematic diagram on the right, we just basically stuck stickers on, and then we discovered, aha, there's some areas, and that's where the antibodies are binding. So that was the reason I was at this particular meeting. And so there was somebody in the audience that I didn't know that kept asking me an awful lot of questions. And I just thought, who is this person? And then I discovered that was Richard Lerner. And Richard had been working on peptides. And he'd been working with Greg Sutcliffe and things with the brain, but they had started working on looking at some peptides from Maloney leukemia virus, and discovered a peptide that didn't seem to fit with the receptor on the surface. And it actually gets cleaved off. But then sort of the idea is you could actually take antibodies to this peptide and actually identify particular antigens, particular binding proteins for these antibodies, and get information about in this particular virus. So that was a little bit, and I knew that Richard was interested in influenza virus, and it wasn't to find out until a little bit later why that was the case. So I was then looking for a job, and I was trying to go back, I thought, to the UK, to Oxford or to London. And it was very hard getting a job back in 1981. There were not many places for X-ray crystallographers, which is what I was. They hadn't gone through this huge expansion that had happened. And so I decided, well, if I can't work back to where I was wanting to go, let me think about the US. So I decided that then I'd look in California. That seemed like a good place. So I managed to arrange seminars at Berkeley, at UCLA, at UCSD, at UCLA, Berkeley, UCSF, UCLA, and UCSD. And Nikki and Richard had come out to actually talk to me a little bit about the hemagglutinin, and I guess I had let it slip that I was coming out to California. And Richard said, well, why don't you come and visit me? And this was a Saturday morning at 8 o'clock. And I thought, good, I'll be in and out there pretty quickly because I want to go to San Diego Zoo. I might never be here again. So I went into Richard's office, which at that time was in the immunology building. It was in this corner office there. And I got in there, and the first thing Richard said to me, I want you to come to Scripps. Now, this was a total shock. This is not what I was expecting. 3 or 4 hours later, and he had a letter. He already had a letter of a job offer. So I went in, and we discussed it, and he kept disappearing, going back and forth. I think he was going back and talked to Frank Dixon. And so we negotiated a new letter with an offer. So I went back totally shocked. So there was me thinking I was going to the zoo, and I got a job offer. So I took that offer back, and it was a pretty bold move to make because this was it. It was a really good immunology department. There was nothing else there. It seemed like a pretty risky move. But Richard was relentless. He phoned me probably almost every day for several months while I was making this decision. And eventually I decided, yes, I would make the move. So I arrived at Scripps in 1982, and that was just shortly after, and I've got a prop here. So this was, sorry, I'm not going to dig up any dirt. So this was the shovel that was used to break ground on this building, molecular biology. And at that time, in 1982, from this, we still didn't have a title for the department. It was called Molecular Genetics Building. And Richard was the chair of molecular genetics, so that seemed like a reasonable thing. So this transitioned into the Department of Molecular Biology. And if you look at it today, you think this was built as one building. It wasn't. It was built as 2 component pieces. So the first part of the building, Richard had got money based on all this peptide work from Johnson & Johnson, and that was the first half of the building. And I moved in here, and there were 6 of us, assistant professors, along here in connecting labs. And as soon as the building went up in 1983, it was built really remarkably quickly, then there was a wall where that line is, that black line is, and Richard managed to convince Pittsburgh Plate and Glass to actually give us more money to form the cell biology department. So that building got expanded, and at the end there, chemistry department wasn't there, it was the vivarium. So on the inside, Richard wanted to do something a little bit different. He wanted to show that we were going into sort of molecular things. So he had known that we had sort of managed to build a model of the hemagglutinin. And so we got this person, John Mack, who had built a couple of these models previously, one for Harvard and one with John Scahill at Mill Hill. And so we got the only intact trimer that was made. And it was made in a case by the machine shop with Ward Coppersmith. And so this is still existing today. This is a picture I took only 2 days ago. And you can see it's not denatured, it's not fallen apart. All of the other models that were made, they no longer exist, I don't think. These parts are very fragile, so as good as a case, nobody can touch them. People always want to touch things, but I have a feeling that some colors are more sensitive than others, that if you touch some of the colors, a particular blue color, it would actually fall apart, but it's still standing. The other thing is that we heard that Richard wanted to build a faculty club, and that faculty club was built in its same size as a lab, because he wasn't quite sure whether he would be able to convince Frank Dixon that we would actually be able to make a faculty club. And this is a recent picture, and I don't know what happened to the picture of Frank, but it's gone. So we started this faculty club, and one of the things I was assigned to do was to find a rug for it. And so I knew nothing about rugs, and so I heard that Leslie Orgel over at the farms knew people that he liked rugs. And so this was an enormous rug to have to buy. And so I found this rug in somebody's garage, and then I came back with pictures of it, and then I had to go over to Arne LaGuardia, he was over in the hospital, and to convince him that we should actually invest in this rug and that we had donations to do so. So that was part of what became a second job for me, was now working in the faculty club, because I actually then had to do all the ordering. You heard about employing Paula Foley. I had to order somebody to bartend, and I did catering on the side. And so, and that was something I was interested in. I did that when I was an undergrad. I was very interested in catering. So I got to spend a lot of time making up menus and doing parties, and especially with Michel Malacot at the French Gourmet, where we had a very good business relationship. So getting back to filling up the building, then on the right-hand side, these were the immunologists. I don't know exactly when this picture was taken, but everybody in that picture, almost everybody in that picture, was an MD. And so we were the new breed. This was the new department. Almost everybody was hired fresh. There were some people that had been here before. And one can see here, for example, Art Olson. Art he was working at Harvard at the same time as me. Jim Hogle, I managed to convince Richard that it wasn't sensible to come out with just one structural biologist, you needed more. And so Jim kindly agreed to come, even though they tried very hard at Harvard to persuade him not to, because they thought he was going to throw his career away. And he had crystals of poliovirus, which he solved a few years later. Greg Sutcliffe was instrumental in these early peptides, of course. He was Richard. And then all of the peptides, it was very hard to make peptides in those days. Bruce Merrifield could make them. So Richard Houghton came down, and he got this tea bag method to actually make peptides. So this was the department. So why was Richard so interested in the hemagglutinin? And that's because Nicky and he had been working on peptides of influenza virus and wanted to make antibodies against these particular peptides. And so he made, Nicky and Richard and the team here worked on making antibodies to these, and found out that they could actually bind the hemagglutinin. They didn't neutralize it, but they actually bound it. But on the other hand, if you raised antibodies to the intact hemagglutinin, they didn't actually bind to the peptides. So there was something that told you something about that the peptides have to adopt some structure, and probably it was a little bit more than just a single peptide that made up the binding sites for many of the antibodies in natural infection. So the first paper I had with Richard, that I was on with Richard, was in 1983, and this was following up on the same peptide angle, and it was peptides from one of these, peptides from flu. And what was intriguing Richard was, well, how could you make an antibody to a peptide which was very flexible? And how could you make it such that it would actually bind to that peptide when it was part of an intact protein? And what was surprising from this paper and the publication is the high frequency for which you could make protein-reactive antibodies that were made against peptides. And in typical Richard form, he decided that this was something that had implications for the protein folding problem, so he expanded that to thinking about how do proteins fold in general. So I came in to do structures of these antibodies with peptides, but it was going to take a while, as you'll see, to actually get a structure. So we actually then thought, well, let's try to figure out what a binding site for an antibody would look like. Now, it might not seem obvious today because we think we know all of that, but back then, the only antibodies that we had were to small molecules. We had no idea how an antibody saw a protein. And so coming up with Art coming in for really expert in computing graphics, Mike Connolly was brought in by Art and started to develop some computer graphics and to actually illustrate this. And so this was completely revolutionary type of representation. And remember when we got the reviews back, they said there was excessive color and too many graphics. So the most important thing about that, what I learned from Richard, was the title of the paper is all important. And we spent hours and hours and hours of thinking about titles. And this one is, you could say it's a little bit of a pretentious title, The Structure of an Antigenic Determinant in Protein. But what did I learn by that? We didn't define what the protein was. We said it was a structure of an antigen determinant. So what did that tell you? That must be the first representation of an antigenic determinant. So that was a real learning experience for me, that the title is all important. And you don't have to say first, you don't have to say novel. The title says it all. So on the basis of that, I started to get invited to meetings, and Raymond has shown a lot of the places that he's been with Richard and many that we've overlapped with. And one of the first places I went with Richard was on the basis of these peptides and thinking of using for vaccines was to Bogota, Colombia. And so there's Richard and I here, and on the right-hand side, I got close, but not talking to the president, but I got close when Richard met the president of Colombia. So that was the first meeting, and that was to become a series of meetings. Then we went again 2 years later, and there's Richard again meeting the president. And so there were two events that happened here that were of great importance. And so Richard came up to me. We were invited for una copa de vino, and simply una copa, that was it, de vino at the palace. And Richard came over and said, the president wants me to sign something, and it's a treaty. He said, we can't do a treaty between Scripps. So he said, what do I do? So I said, not much else you can do, sign it. So there was signing a treaty between Colombia and Scripps. So the reason that Richard had got to know the president quite well from that previous visit, and on the right-hand side, we can see a photograph, and the person over here is actually the most important part for me going down there. That was meeting Jairo Arevalo. And Jairo was a student that worked down in the labs there for Manuel Padroya. And you can see that with Art, Jim Hogel, Peter Wright. And I had said to Richard, wow, there's an incredible amount of talented people. And Jairo was assigned to me. We were assigned a chaperone to make sure we didn't get into trouble, I think, more than anything else. And I was really impressed with Jairo. And I thought, well, this would be terrific if we get people like Jairo to Scripps. So Richard managed to get word to the president, and the president announced at the end of the Una Copa de Vino that he was going to have a presidential fellowship, and Jairo was going to be the first presidential fellow. So Jairo arrived in 1985 with $10,000 in hand in Florida, which was a pretty dangerous thing to do, with the money that was supposed to pay for the fellowship. That was the last we saw of the money, but that was we'll see why Jairo becomes important, even more important later on. So as a side thing to these meetings, and the last one I'll show on these trips, was that I got invited to Cartagena a few years later. And thanks to this meetings setting up and going down there with Richard, I kept getting invited. And so I was able to meet a true visionary. This is Juan Valdez, the spokesman or the representation for the coffee from Colombia. And so I managed to get a picture with him. This was the real one. He had doubles. He was so famous, he had doubles. And I was assured that this was the real Juan Valdez. And I suppose that was true because it's the only time I've appeared in the front cover of a national newspaper and with Juan Valdez. So that was somebody really important that I obviously met. So getting back to the science, Richard was still intrigued with, again, you know, what does a peptide sequence look like in a protein? And can the same sequence adopt the same or different structures? And because if you were going to raise antibodies against them, what if a particular sequence could adopt two different structures? Then would these antibodies be able to differentiate between these peptides in a protein? And in fact, we found, not surprisingly, that there weren't many sequences out there in those days, and there weren't many structures out there. So we found up to, up to I think it's 7 MERS, it's occasionally 8 MERS, but they were only the protein dated back up to 7 MERS of sequences that were very similar to one another. But quite surprisingly, we found the same sequence had completely different conformations in proteins. And that again said about this idea of what is an antigenic determinant, what does a protein, what does an antibody see when it recognizes a protein? So I set out and looking at trying to set up crystals of antibodies. And I'd been told that they were very difficult to crystallize. Roberto Polyak, one of the pioneers in this field, told me only 1 in 25 to 1 in 40 antibodies would crystallize. David Davies was a lot more optimistic, 1 in 5 to 1 in 10. So I thought, well, I'm not going to put all my money on one antibody or one set of antibodies. So we started looking at a lot of different antibodies. And so Richard encouraged me to actually pick up more different antibody projects. Another project with him was an antibodies to my hemerythrin. He put me in touch with Arnold Feinstein. We started working on IgM, trying to solve the whole structure of IgM. But fortunately, he had some antibodies to progesterone that we could make FABs on. And so I can assure you that Richard looked at every one of these single crystals. Every time we had a crystal, he came across and looked at it. And it took, and we also started working on some other things. It was another crazy thing. He just encouraged us to work on whatever we wanted and to really think big. We worked on IL-2 receptor. Here's some crystals of that in the middle panel here. We worked on LDL receptor with Browning-Goldstein. Worked on rabies virus glycoprotein with Hilary Kraprowski. Worked on MHC2 with Ralph Reisfeld. Worked on just enormous number of things, some of which took 20 years later to have the technology to solve. But we were into growing crystals, and that was, for us, that was the beauty. We're much better, when I look at it, we're much better at photographing crystals back then than we are now. So solving structures was quite hard in those days, and even with FABs that you think we can solve very quickly now, we can do in a matter of hours. But there was no way of solving an FAB structure by molecular replacement, which is the quickest way to do it. But that changed at the end of the 1980s, and we eventually got the first crystal structure. Well, you can see I came in 1982, and this is 1991. If it had been nowadays, I would have been kicked out. Richard kept the faith, and I managed to survive through all of that. And we produced this first structure. So this was another peptide antigen from my hemerythrin. And you can see the peptide nicely nestling in here in a groove in the antibody structure. then the structure started to come fast and furious. And what I had come to Scripps to work on was these antibodies to influenza virus peptides. And here was our first peptide structure. That fancy graphics I showed you with it, it was from a peptide from that particular binding site. And so we could see again that the peptide fitting into a groove and the antibody peptide sitting in the middle there. And what was interesting about it was, if you forget this pink is here, but we just put this in, this is a form without the peptide bound, there's no space for this. So that gave us, there was still a lot of question of whether antibodies bound like lock and key or induced fit. And so here's another one of the Richard titles, Structural Evidence for Induced Fit as a Mechanism for Antibody-Antigen Recognition. It doesn't say what the antibody is, what the antigen is. It just illustrating this is one of the first examples for induced fit. What was also interesting about this peptide is that you could put it on the end of another protein as a tag. And so many of you know this particular peptide is the HA tag. And so this is what you can buy antibodies for. You can tag your protein, pull out that protein using that particular tag. So that was something that was very useful that came out of those antibodies. Jairo, that I told you about, had come to work in the lab in 1985, and then in 1993, he actually was working. He had solved structures with antibodies to progesterone and derivatives. And if you look at the right-hand side, you think all of these, superficially are very similar to one another, but they're actually all different shapes. So even though they're small molecules, some of them are flat, some of them are bent, and some of them are even more bent. So how can an antibody bind to all of this? And so that was a series of structures. This is the unliganded form. And you can see here's one of the derivatives, here's another one, and here's another one. So they were using different pockets. So this one came out here, left that pocket free. This one moved into this one, left that pocket free. And in fact, in this one, which is the most bent one, it basically flips around and goes upside down. So this was really sort of gave us some insights as to how well antibodies can actually fit around something and why there's limits on the complementarity to the interaction. And this also showed us some induced fit because you can see without the ligand there's a tryptophan here, and that tryptophan flips around and comes over to here, again creating a space for the ligand to bind. So by this time, Jairo, he came to my lab as a postdoc, but he now was a graduate student. So this is odd. So he decided that he wanted to actually follow an academic career, and he had done 1 year in Columbia, and he really didn't feel he had a proper degree. So he became the first student who graduated from the graduate program that we heard about. So Richard decided that we should have a commencement ceremony just for Jairo. And so here's Jairo in the middle here, me, and then the founding members of the graduate program, Bernie Galula, Bill Beers, obviously Richard, and a member of the board. So that was a very, very special occasion. And that enabled our program to get accredited. And so everybody else after that could actually officially have an accredited Scripps degree once Jairo graduated. So we've heard about catalytic antibodies. There's sort of joint papers back to back in Science, 1984, the Wolf Prize, the Ehrlich and Danster Prize, which is shown on the right with Pete and Richard. And so that was really, it was very exciting for me because I had worked on enzymes when I was a a graduate student. And I was so glad to go back to thinking about enzymes again. And so combining antibodies and enzymatic activity was great. So it started on, this is only a small number. You've seen some of these images before. There are unshown some of them. And so you can see that we actually started to uncover how it was that antibodies that could bind to things could actually catalyze reactions and have part of the activities were very similar to what natural enzymes are doing. Up in the middle portion, the lysine, for example, that was a very important residue that got activated that could take part in the catalytic mechanism. We could see that there were reactions for which there were no natural enzymes, and were even quite difficult to do by synthetic organic chemistry. Maybe not for Phil, but for most people. And so this was really a wonderful time actually exploring what antibodies could do other than bind things and actually make new compounds by using these particular antibodies. We've heard about Richard collecting things, and so he also collected samples. And so back in 1973, a patient was diagnosed with multiple myeloma. She was very yellow, and the antibodies were collected from her, and these antibodies are shown on the right-hand side there, and you can see they're very, very yellow, so it indicated they were binding something, and it was thought to be a cofactor. So 25 years later, Richard remembered he had this stored in the freezer and pulled out this tube and came to me and said, "Can you solve the crystal structure of this?" And I thought, oh God, we've only got one shot at it. This is the only sample. What if I destroy it? So we had to, it was an intact antibody molecule. We had to cleave it into the component binding part, which is the FAB. We managed to do that. And in fact, we found that riboflavin was actually bound to this particular antibody. And so, again, Richard was always amazing because he wasn't just thinking forward, he was thinking back on problems that hadn't yet been solved. And here was one that was sitting in the freezer, and we were fortunate to be able to get a structure. And that led to the idea of thinking about, well, antibodies could have cofactors in their activity as well as whether to bind or in catalysis. So we hadn't forgotten flu, and Richard hadn't forgotten it. So, more than 20 years after I went to Scripps, he actually had an opportunity to get some antibodies from an H5, the bird flu infection, back in about 2007. And so he actually made a library of antibodies from a person that had recovered from influenza virus. And he was the first person to publish on antibodies that could cross-react between different subtypes. Now, this was very rare, very unexpected, because usually with influenza virus, you get very specific antibodies only to that particular strain that you're infected with. And here, Richard had pulled out antibodies in collaboration with Celane that could actually bind not only to different antibodies within that subtype, but also antibodies between multiple subtypes within influenza A viruses. So here's the groups that are in the top. This is influenza A virus that could be represented by 16 different subtypes. And here was some antibodies that could bind all of that green group. Now, this was astonishing, that you could actually find something this broad. It wasn't thought that human antibodies were able to do that. As with most major discoveries, shortly after that, there were papers from Crucell and papers from Wayne Marasco in Boston that identified similar types of antibodies. And what was similar about them was their sequences. And so they were all from a particular germline family. They were all highly related to one another. They had come out of patients in different places, different continents. And the features that they had that seemed to be important was the actual germline features. They had some somatic mutations. There weren't many. But it gave us an indication that this region was important for the binding. This particular one of the 6 loops of an antibody that actually grab hold of a protein. So we set out about looking at the crystal structure of these to see where they actually bound. And we had expected that there would be, that most of the antibodies that we knew then, I don't know what we expected, but most of the antibodies that we knew then were binding to the top part of the molecule where most of the variation was going. However, these antibodies, this is one Crucell, this is one from Richard and Seelane, here's another one from Crucell, and this is one that we didn't solve from Marasco. And you can see they all bound to the same region. That was really amazing that these antibodies were pulled out and they all had very similar features. And here's, if we look at in here, then this particular loop, that previous sequence I showed you, here are the key residues, an isoleucine and a phenylalanine, and there's only one family of antibodies in the entire immune repertoire in humans that has two hydrophobic residues at the tip there, and they were burying these hydrophobic residues in a pocket. So everything sort of moved pretty fast after that. So antibodies were found to go these were antibodies that were bound to, as I say, 10 of the subtypes of influenza A. Antonio Lanzavecchia found antibodies that bound to all of influenza A. And then in the collaboration that we had with Crucell, they found another antibody that could bind to both influenza A and B. And so these were antibodies from people who'd been naturally infected or vaccinated, and here's ones just from vaccine. And what all of these antibodies were doing were basically binding there, and they were sticking pieces of the antibody side chains, the amino acid side chains, into pockets. And these 3 pockets that were most important were below, that were filled with an isoleucine, a phenylalanine, and a tyrosine. So I'm not going to go through the whole story of this, but then we sort of got interested, well, could you actually make a small molecule therapeutic? And this was not the way that you would think of doing that. You think of screening small molecules against the protein, but that had been totally unsuccessful against the hemagglutinin. It was thought to be undruggable. So we thought, well, can how the antibodies bind help us in designing therapeutics? So in a collaboration with David Baker, just after the first structure came out, he decided that he could make this small protein molecule that could bind in the same site of the antibody and inhibit influenza virus. He then made another one which is smaller and another one which is even smaller. We thought, well, could we actually do something even smaller? Could we go to a peptide? And so, again, with Crucell and now Johnson & Johnson, taken over Crucell, made a small peptide that could actually bind and also inhibit. And then the last thing was, well, could we get a true bona fide small molecule? And so by screening against these smaller molecules of the library, then a small molecule was pulled out that was, you can see, way much smaller from this 46 kilodalton antibody now down to a half a kilodalton small molecule. And we followed this up with Dennis Burton, and we've now got compounds. This is one of the compounds with Dennis. And we've got ones that are binding in the low nanomolar range. So that was for therapeutics. So that whole discovery led to ideas of now how you could target this molecule, this protein from influenza virus with small molecules. And so what about vaccines? Well, most of the antibody response in that original picture I showed you of the hemagglutinin with the dots on it, that's where the antibody response is. It's the so-called immunodominant sites. And so we thought, well, we want to get rid of this, so we want to chop the head off the hemagglutinin. And it's easier said than done. To get this to stabilize its two chains, it was quite difficult, again, a collaboration with Crucell. And so we managed to get something that we could actually now inject that small piece of the hemagglutinin made as a single peptide and induce antibodies against it. And these antibodies were much broader than anything you would have got with the intact hemagglutinin. So that gave us an idea of this could be a target for universal flu vaccine and help us in that regard. So Richard wasn't yet finished with antibodies to the hemagglutinin, and so he pulled out another really interesting antibody, again in collaboration with Celine. And this antibody was very unusual in the sense of it had some breadth, which we didn't expect for something that bound to the top. I've told you this is hypervariable. This is very conserved. That's why these antibodies can have much more breadth. And so when we actually solved the structure of this antibody, it had a single binding loop. So it has possibility of 6 loops to bind. It only used one of those. And why is that significant? Because you're trying to inhibit the receptor. The receptor is sialic acid and some other sugars. And so if you put a single loop in here, then, and don't touch the sites, which are variable, then you can basically specifically target this site with an antibody. And so a number of other antibodies, we worked on other antibodies. There's some of them are shown on the right-hand side here. And actually figured out that how you targeted that was you basically put an aspartic acid down here and a hydrophobic residue, mainly an aromatic over here. And so that gave some idea about how to target that. And David Baker followed up on that by basically taking that loop off this antibody that we got from Richard and Celine, cleaving that off, sticking it onto another protein, and using that as an immunogen. And basically in the bottom right there, I don't have time to go through this whole slide, we can see that that was very effective at inhibiting, as a general inhibitor, of the receptor binding site. So that would inhibit viral entry. The last structure, the last set of structure sites will come back. Well, let me talk about this one. You've seen this slide a few times now. And we've solved quite a number of structures against SARS-CoV-2. And these were isolated from people who had been infected or vaccinated. And so what Richard wanted to understand was he had a library that he had made 20 years ago. Would any antibodies in that library see SARS-CoV-2? Now, it's a large library, so you'd expect some antibodies to bind. But these antibodies that got pulled out, they had a number of somatic mutations, and they bound to the receptor binding domain, which is that little magenta thing on the spike protein of the virus above. And so they bound specifically to the receptor binding site and blocked interaction with this receptor. So the question is, and this library was made from a mixture of antibodies from different people, the combination of antibodies from different people. And so Richard was very intrigued in this, is where do these antibodies come from? What was the antigen that they saw? Was it a virus? Was it something else? And so this led to some ideas about thinking about, well, was there something that pre-existed before the current SARS-CoV-2 that looked like it, that these antibodies were made against by one of these people? So this is still something we don't really know, but it was an intriguing observation. And this was the last paper that published with Richard that Raymond also showed, and a collaboration on 3 continents. And this was, the antibodies were pulled out by Guang Yang at Shanghai Tech. So let me finish by returning to this slide. So the whole new era of Scripps was founded on synthetic peptides, and particularly on using as vaccines. And so I haven't told you so far about antibodies that can be made to a peptide that can be used as a vaccine. So let me sort of tell you two instances of where peptides are being used as vaccines. And this is linear epitopes to HIV and to SARS-CoV-2. And right down at the bottom part here, as the spike protein, this is the whole spike protein, as it enters the membrane, there's a linear peptide sequence here, and antibodies can get at that particular site. The same is true with, that's with HIV. The same is true with SARS-CoV-2. In a very similar region, there's some linear sequences here, and antibodies can actually get at that. And so what I show you in the center here are structures that were done in the lab showing these linear peptides binding to antibodies that were produced, that were elicited against either HIV or SARS-CoV-2. This is the HIV one, and this is the SARS-CoV-2. And just parenthetically, if you get that close to the membrane, you're also going to have to pick up some interactions with lipids. The second site is good for thinking about targeting with linear epitopes is the fusion peptide. So all of these antigens, all of these viral spike proteins are made as a single protein, and then they get cleaved to release the so-called fusion peptide that can actually insert into one of our cell membranes to enable the viral membrane to fuse with the host cell and get the genetic material inserted into the cell for the virus to be able to replicate. So this fusion peptide, as it turns out, is not accessible in the hemagglutinin, but it is accessible in HIV, and it's also accessible on SARS-CoV-2. And again, coming back to where we first started, here we have antibodies to peptides that actually are, give us hope for much broader vaccines, and for HIV, it's a very challenging vaccine to get. We still don't have it. And that's because basically you've got thousands and thousands of different strains circulating all at one time. When you get infected, you keep making new viruses within you. And so it's a sort of an arms race with the immune system. But it has to have some conservation of regions, and it has regions, in this case, I showed you in the base, and also over here, that are involved in the fusion machinery, and these look like promising targets. So, since I came to Scripps, I first published with Richard in 1983, and the last paper was this year. So it's been continuous. So he's impacted my career, my lab immensely over the years. I don't know where I would have been if I hadn't started. I was interested in influenza virus, but I had no thoughts of working on peptides. I had no thoughts of working on antibodies. And so that's led to all kinds of things that we've been able to do in the lab. And Richard was always there thinking of new things to do. I haven't shown you some of the many things. He would come in, he would phone you up and he'd say, I've got this idea. And you'd think, you'd tell it to you, and I said, yes, let me think about it. And I have to go back and read something because I had no idea what he was talking about. I said to Richard, where do you come from? Where do you come up with all this? It's just amazing. I don't know how somebody can think of so many things to do. And he would go backwards as far as forwards. We heard about Goodpasture's disease. He came back to worrying about Goodpasture's disease. And so this has been, I just don't know how we've done. I've never, ever wanted to work anyplace else after I came here. Richard was just so incredibly supportive in good times and in bad times. And he was just an enormous friend. And I think excuse me, I had to try to leave all this to the end. I knew one of the things that I think I would like to leave you with is something which one of my former students wrote after hearing about Richard's dying, and said he was obviously sorry to hear about Richard. I took for granted how we'd come to our graphics room to look at our new structures. Later I learned nobody really does that. He really cared about other people's success. We were like his big extended family. That's what I want to finish on. Thank you.
Thanks, Ian. I think we've all heard today that Richard was not only an amazing scientist, he was an amazing builder, and he was really an amazing person. And when I took on the presidency of Scripps, they're really huge shoes to fill. And so every now and then I would go to Richard, even after his stroke, and ask Richard for advice. And Richard was always very succinct. He would say, do it. Do it. And then finally, Richard, where should we go? And he was like, Pete, you got to pursue really big ideas. Always. So that was Richard. As Phil said, Richard's spirit's probably here. So why don't you all join me in giving him one last tribute. And as Richard would have loved, please all join us outside and share Richard's stories with your friends. Thank you.